think my slide sharing should work. So welcome everyone. This is our second joint retreat with between Michigan, Pittsburgh, Weiss, and CDOCTR, as we're in year 2 of the stage 3 proposal. So on behalf of the leadership and everyone at both resource centers, I want to welcome you to our retreat. I think most of you are aware of the great partnerships that exist. For those of you that may be attending for the first time, both resource centers are really a strong conglomeration of very good institutions from around the country. And beyond that, we have a lot of advisors and partners and experts, not just scientific and clinical, but really the expert machinery that it takes to take a project off the bench and get it to the clinic. We want to welcome and acknowledge those people, as well as the support of the NIDCR through first 2 and currently in the 3rd stage of this initiative of NIDCR. I won't call out all of the leadership, just give an acknowledgement en masse of the leadership from both resource centers as well as our external advisors, and tell you that the mission of this program, of this initiative, is to really be the premier resource center in the US for taking doc therapies, doc technologies, and catalyzing their translation through the pipeline, and ultimately not just into patients, but true clinical adoption. This slide is the machinery of the overall NIDCR program that was envisioned by Martha Summerman and others probably 6 or 7 years ago now. The first stage was a 1-year planning grant. Teams self-assembled, and CDOCTR and us had independent resource centers in stage 2, in which we recruited and vetted and funded a number of ITP projects. And now in stage 3, it's really a consortium and a true partnership between the 2 resource centers and the NIDCR. So we are about 1.5 years into stage 2, probably into stage 3 rather, probably 5.5 years into the total 10-year consortium that NIDCR envisioned many years ago now. So for today and tomorrow, at a glance, I'll hand things over to Rena for a more extensive set of welcoming remarks. And then tomorrow morning, Yang and I will give a consortium update in more detail than I'm able to give now. The bulk of the time today will be progress presentations from the ITP teams from both resource centers. And then the last few hours this afternoon will be a breakout session under a general topic of critical considerations for preclinical product development. But we've bucketed the breakouts by clinical indication, and each indication will have a number of discussion questions relative to regulatory issues, product development issues, marketing issues, commercialization, et cetera. And this is a great opportunity for the ITP teams and the overall membership in both resource centers to put their heads together, talk about best practices, what's worked, what we might need help on, and what we could tweak to make things go even better than they are. And then tomorrow, the balance of the ITP presentations in the open session, a keynote talk from Laura Silverman on translating technologies to the clinic. And then we'll adjourn, and those of us in leadership, our external advisors, and our NIDCR partners will have a closed-door discussion on the progress and any funding decisions for the ITP. So that's the organization of events for today and tomorrow. What we'd like everyone to get out of the retreat is an understanding of the ITP teams of the two resource centers, have an update assessment as well as a joint review of progress. The breakout session should be both educational as well as helping ITP teams in directing their technologies through the pipeline, translational pipeline, and then a little bit of strategic planning following the closed door session tomorrow afternoon. So with those brief outline, again, Yang and I will do a more substantive update, State of the Union, so to speak, tomorrow. This is the agenda and the objectives. And I'm happy now to introduce Rena D'Souza, the director of NIDCR, to give a keynote talk. And it's really the vision of the NIDCR that created this program in looking at R01s being funded over the past 25 years and what could we do better to create more translation. So thanks to the vision and all of the leadership of NIDCR, now I'm happy to hand things off to Rena for her presentation.
Can I share screen? I could. Okay, so I bring many warm greetings to all of you and so much gratitude for and appreciation for you keeping the community connected in these times. I urge you to continue to have the finger on the pulse, especially for women and caretakers who struggle with individual circumstances that are challenging. So do let us know, because NIDCR staff is very responsive to those extenuating or just simply timely circumstantial challenges. I want to thank Lillian Shum and her program staff who always keep me well informed of the progress of DOCTRC and all that's happening. So what I really want to do, and I have this picture, my favorite sculpture from Rodin, is to share with you in really a non-prescriptive way, and I really mean that, a landscape of what's happening in the world, of biomedical research, of regenerative medicine, as the pandemic has shaped our thinking and our culture in so many ways. And hopefully from these this overview, there would be food for thought amongst the leaders as we project for the future as to what should DOCTRC look like 5 years from now or a few years from now. So what I want to do is to sketch out where what we've done at NIDCR, just a few of the initiatives that are in place that somehow magically occurred in this virtual reality we lived in, to talk about the pandemic and the paradigm shifts that is created for biomedical and health research, emphasizing two particular programs that I think herald a new era of discovery for us as we move into the age of what they call use-driven research, not discarding or ignoring the curiosity-driven research, but making sure that the patient stays at the center of our inquiry, because we are in fact the National Institutes of Health. And that would hopefully lead to a planning for the future for DOCTRC, which we will count on all of you, your leadership, your ideas, and your experiences to guide NIDCR to the next phase. And I put this out there because I really do feel the stars are aligned now like never before. So I bring with this a sense of hope and optimism for the future. So this is real quick, great accomplishment for this year, having two brand new dentist scientists. Renee Jasko, in fact, is not quite brand new, but coming home as captain, a lot of experience in HRSA, FDA, worked extensively at NIH with CTSAs, etc. Very well networked in the community, very interested in T1 to T4 and actually community translation as well. So Renee joined our staff as senior advisor and will be actually directing the public health residency program that is now shaped to be 1 year plus 2 years of an intense postdoc research experience. And I'll go into that later. And I'm thrilled to introduce Jennifer Webster-Syriac, our new deputy director, first day at work today. So I asked Jennifer to join us. Jennifer, do you want to say hi? And I can I can brag about you in a minute if you are able to. Are you?
Certainly.
Oh, yeah.
Certainly. I'm so glad to be here today, and what a wonderful way to start my career here at NIDCR, learning about the very important work of translating what we've been doing out to people, because we know a lot, and there are major opportunities to decrease oral disease burden by doing what you're doing. So I appreciate what you're doing with DOCTRC and look forward to learning more about it. Thank you for having me.
Thank you, Jen. So as a dentist scientist, Jennifer comes from the viral pathogenesis field, also very keen on setting for NIDCR a direction for health disparities research on campus, as well as engaging extramural investigators. So happy to have you. Okay, bragging point number 2 is this. 15 years ago, David Julius came to us and with projects in an area that was not well explored at that time to really look at nociception, taste, and thermal receptors using capsaicin. David received 8 years worth of funding from NIDCR approximately, and then Autumn, who is also a Nobel laureate along with David, was funded for 15 years. And it's great that they were both, their foundational work actually occurred because of the support of NIDCR. So when I heard the news, I immediately called Hal Slavkin and Larry Tabak and thanked them for that wisdom in investing in an area that is so important to us as human beings, being able to sense our environment and react to it. And COVID certainly has shown that. So here are 2 bragging points. So our strategic plan is in its final stages of editing right now. Take-home message, it's going to be focused on translation and not looking at it as a linear continuum between basic and clinical, but rather a circular continuum with the patient in the center. Knowing that we have to have a workforce that in fact matches our demographics on so many levels is important, and we're taking that very seriously. And also importantly, looking at how we measure success and how we define it in the first place, and then measure it so that we know to keep what works and to maybe move along or improve things that don't. So that I think really is the hallmark of the strategic era we moved into. And these are the 4 goals. And I put this from NCATS as being significant here to signify the continuum, the importance of foundational research, but the relevance to the patient in the center is an important, succession plan that we ought to have for the next era that we enter. And we move from oral systemic connections, actually putting meat on the bones in terms of cellular molecular determinants, recognizing that the oral cavity is the portal of entry for nutrition, for e-cigarettes and tobacco, for actually being the window to the rest of the body in so many ways, being able to monitor diseases and the success of our treatments as well, our systemic diseases as well. Moving on then to take that knowledge and try to transcribe it in a very personal way to individuals. And we do have certainly the resources available at our fingertips today to do that. Implementation sciences lends itself to a great scope of development in these years to come, being able to take our knowledge and transcribe it to practices for dentists and oral health professionals in general, and of course our communities. Training a diverse group of oral health scientists who are really well versed in an interdisciplinary way to take advantage of emergent opportunities. And of course, expanding our existing partnerships. And you'll see examples that we have, that I'll share with you, that actually signify progress along these routes. I thank all of you that gave us input. There was extensive feedback from council and from the public. So thank you very much. From there, for those of you that sat through the last council, you'd know that there were 10 concept clearances. It was pretty intense. And we continue to put out notices and funding opportunities so that we can move things along in the right path. So even though the plan is not released, we certainly are moving in the right direction in many ways. Much awaited is the report that was originally commissioned by the previous Surgeon General, but then became the responsibility of NIH and NIDCR. We did put forth the funding for this report that sought contributions from so many experts in the field. It's truly the voice of the community, but it's not meant to sit on a shelf. And so we have, Renee will be in charge and working to disseminate it wisely, to look at calls for action and make sure that it has the maximum impact it can have, both on oral health in the US as well as the global scene as well. There are two short perspective articles, one in the New England Journal of Medicine and the other in the Lancet, that would herald the report. It's due to be out in December, so it will be a 21 gift to the nation. It was quite an experience, actually, kind of guiding it through with the help of staff through government layers of the government, all of which I think turned out to be a great experience for me. So much of the report actually highlights how important the systems we engage in are, but they also bring with them unique challenges that I don't think are felt in many other systems in the body. We have such tiny spaces to work in, small tissues, unique microenvironments. All of these allow us to actually showcase paradigms for organogenesis and for tissue homeostasis and regeneration. You're very involved in all of that. But we have this is actually what is glaring us, staring us in the face. We have very hard numbers to justify when we talk to Congress. We still, 50% of all craniofacial, all genetic disorders involve the craniofacial complex. We have the most prevalent childhood disease of mankind, caries. There's progress in each area. Periodontitis still claims so many in this country, leading to sometimes loss of teeth, edentulosum and cognitive decline. And oropharyngeal cancer, especially the HPV positive ones, are now dominating, actually superseding cervical cancers. The vaccine bringing us much hope, but there's also a lot of hope for diagnostics and biosensors. Based on public health needs, we decided to revive the dental public health residency program for a year, tagging on 2 years of postdoctoral research in very cutting edge areas on campus. NCI has a fantastic epidemiology program. For example, the National Institutes of Aging are very concerned about aging, cognitive decline and edentulism. We have the National Library of Medicine that offers huge data science and bioinformatics programs that our trainees can avail of, so that we begin to train a cadre of research-oriented public health dentists. And we realized during the pandemic that we really don't have a robust pipeline. There are great examples of experts, Harvard School of Dental Medicine, for example, leading the way in so many areas. So institutions have it, but we probably don't have enough, given what we face today. What has been sitting on the burner since the Academy's report, but a lot of work behind the scenes, engaging a multi-council group of thought leaders, as well as extramural folks, to create a consortium for temporomandibular joint disorders, requiring, as you can see here, truly a multi-pronged approach. And we're now in the process. Lillian Shum is going to lead that initiative with other institutes on campus. We have several who have a stake in all of this that will come with us to kind of solve the problems in a very kind of systemic approach way, systems approach way. So watch out for that in the years to come, months to come, sorry. We've realized, not just reading the report, but over time that we can't really cure or treat a given disease without looking at the upstream health determinants that shape an individual, family, or a community. And for that reason, we are, and Jennifer is going to play a major role in setting up a presence for health disparities based on individual, family, and community level influences. I'm going to go through this real fast. I've put this out before. It's my favorite slide because it really talks about the importance of connections, both globally and nationwide for us, that there's never been a time when innovation ingenuity and integration. So these are the I's from immunity, immunology, and infection that are really kind of creating for us a new paradigm on how we perform research in a manner that is meaningful to our society. We know that science and technology has won, and will continue to, with the president actually having Eric Lander on his in his cabinet is one step in that right direction. I was privileged to have met him when he came to campus this last week, NIH, and he talked about his 5-point plan for the pandemic. And it's clear that he respects science and everything that went behind the creation of the vaccines and the diagnostics over this period. So I want to thank you when it comes down to leadership. Thank you all for keeping this going in this time. An example of some of the technologies, one is the smart mass that is, I think, going to be useful in detecting proteases on the surface of viral exposure. So that's in the works. And I threw this out just as an indication of the kinds of technologies that RADx and Active have created for our field, many of which can actually continue to linger and be translated to all the problems that we in oral health have encountered. And this is just one example of the home test kits available, lots of patient-centered technologies that can be used to our advantage. We do have a very large grant right now looking at recovery and what happens in post-acute sequelae of COVID, some of which actually do involve oral symptoms. That grant, that huge grant is running as a consortium nationwide, is based at NYU. Now, what is interesting, I think Young Chai and David, you'd be curious about this, and I'll feed more information as it comes about. So Bruce Stolberg, who is the director of NIBIB, set up a very special mechanism with FDA to fast track technologies. Now, this is particularly related to COVID, to the diagnostics and tests, but I think it heralds a way for which in the future we can streamline and get FDA approved and certainly EUAs for important drugs and therapies. It won't, even devices, because that's what this particular initiative is encouraging. And it's called, what is it called? Gosh, someone help me here. The Independent Treatment Assessment Program, ITAP, and it's right here on this slide. I'll make sure that you have a copy of this presentation to refer to this website. Now, throughout the past year, there's been a tremendous effort on our campus, a very real effort that makes directors and leaders of NIH accountable for our biomedical research community diversity as well as in-house. And this manuscript in Cell really highlights the key activities of this group and what is to come. We also have an example of how weaknesses, and they were really weaknesses and limitations in our clinical trials infrastructure nationwide, that at the time of most need were not really mobilizable. And that has opened up our eyes to the needs to have in place infrastructure that connects our teams with community leaders and church and religious leaders as well. And so this program SEAL is one that is Eliseo Perez-Stadler, who is director of the NIMHD, the Minority Health Disparities Institute, is working on to great success. It also kind of made us think about clinical trials infrastructure as it relates to a national scene. We don't have a national health service like the UK does. And we certainly, like South Korea, couldn't track individuals through contact tracing because we just didn't have the infrastructure. This is all being built in place right now to just handle the rest of the pandemic and also whatever comes to us in years to come. Now, despite the fact that we have a very dynamic set of programs on our campus that are trans-NIH, and NIDCR is involved in every one of them. We have funding and we put down funding, and we also have grants that are awards given through these mechanisms. and especially HEAL, which is designed to really look at pain management and opioids as well. And I bring this up for DOCTRC, especially because of the news that the president's budget, as well as the Senate's budget now, has approved for NIDCR an $18 million increase in funding that will be added to our base, which is great. So it takes us over the $500 million mark, but it's dedicated to pain and opioid research. And we have many areas of dental pain, cranioorofacial pain that we still have no answers for, right? And so we are right now gathering ideas, so please send them along to us. Certainly in regeneration, the control of pain becomes a very important issue, both in delivery systems and scaffolds and means by which we can control or balance out cytokine storms with resolution phases of inflammation and healing. So I think we can all avail of this important new addition to our budget. Now, for some of you that actually heard council the last time, there was this, I invited Tara Schwetz, at that time detailed to the White House, so she was working with Eric Lander, to develop this new initiative. And I bring it up here for you as an indication of how paradigm shifts are occurring as we speak. And she presented this, this fact that we are in an era where cancer immunotherapies and vaccines were developed at record pace, asking, could we perhaps get out of that dead valley zone to accelerate the pace of breakthroughs? Now, this is something that DOCTRC has taken very seriously. How do we really use this sense of urgency to handle society's biggest diseases today, Alzheimer's and others? And can we transform healthcare access, equity, and quality and all of that through new science and technology? And so dreaming if we could develop mRNA vaccines to prevent cancers, can we create molecular zip codes? Can we have platform-based technologies? And can we build user-friendly point-of-care devices that involve patients and allow patients to take ownership of health? And so Biden made that announcement. We were all excited about it. We had, certainly had news at council from Tara. But because of all that's going on with the reconciliation and Build Back Better bills, this got kind of sideswiped a bit, and it is now considered part of the Cures 2.0 Act. Remember, we had 1.0 that dealt with the cancer moonshot and the brain initiative. Well, there's going to be a second generation Cures initiative, and ARPA-H will be packaged within that and at a reduced budget. But it is heralding, I think, a need for us to be moving research along those lines. And so this was what it was meant to, really to transform health through these fast-tracking breakthroughs that work and can be readily accomplished that cannot be accomplished through traditional means. And NIH does still fund traditional research and commercial activity. And so the goals were just like the way they were, as described here. And it brings to mind the fact that these opportunities are going to come along in different ways from NIH, even beyond ARPA-H. And what is driving the culture is the nimbleness, the risk-taking, the fact that we can evaluate things that work early on, drop them, allow them to be supported by other mechanisms. So that's fail early and accept risk and be accountable through milestones and independence. Now so you put that aside, then comes this set of initiatives that I have completely been drawn to. And it made me think, every time I listened to this, and this was just a few days ago, the executive committee update on progress through AMP, I think about the need for us to actually employ a much more in-depth, disease-focused systems approach than what we are currently doing. So if I had to ask the periodontal researchers, do we really have a full sense of what happens in periodontal disease? Have we deep phenotyped the condition? Have we stratified the disease, monitored the progress? did we have the tools to do that in the past, and how can we avail of current tools? This initiative comes to mind. And I just wanted to scan through these real quickly. They target all the major complex compound diseases, tough things that we never thought we would make progress on, have moved at a dramatic pace right now. And it's because of the partnership with industry, with government, academia, and of course, NIH. And you can see, I'll have to go through this, sorry. It's really, the first one was diabetes type 2, then came Parkinson's, Alzheimer's, congestive heart failure with ejection fraction preserved, schizophrenia, etc. And you can see that the goals are very, very parallel, very similar to what DOCTRC has set, but the approach is different. So for example, in the autoimmune disorders category, the first focus was on rheumatoid arthritis and systemic lupus. What they chose to do was to first of all, get this group of folks together, the leaders in the field, along with industry that had very strict timelines, and they deconstructed tissues of significance to these two diseases, looked at single cell profiling, multi-scope analysis, put pathways together, fed this into a bioinformatics, into a data operating system, and came up with targets and drugs that could be used. And you can see here that this involved the transfer of knowledge along the spectrum within this domain of interest, but also there was a lot of crosstalk that occurred throughout all of AMP's initiatives. So things that were happening in Alzheimer's were now applicable, as you can see here, very similar to the knowledge portal created for Alzheimer's. That same model system was applied to this particular approach. Now, because of the advances and the progress made with these two diseases, it was really great to have Sjogren's come on board. So that happened shortly after I arrived, and NIDCR became a partner. We contributed our funds along with NIAID and NIAMS. And we have, as you might know, an RFA that went out and a very, very competitive set of grants and awards to be soon made, using the same principles of reconstruction and having key deliverables in a short period of time. But it involves a thorough analysis of a disease in humans. And most of the biosamples, it's not that they don't have animal models, they do, but the biorepositories are really samples collected from these patients, blood and saliva and tissue samples themselves. So after you deconstruct, you kind of reconstruct in the in vivo context so that you get a sense for, you validate what you found, and you also kind of put the omics together in what they call is the interactome, which is all of these omics that you've heard of. And it sounds very surrealistic, but it's actually happening. And we should see great progress in Sjogren's. And so the question to ask is, in the next phase of DOCTRC, should we be thinking of an in-depth systems approach so that our regenerative strategies are based on really the knowledge of the complete picture, of the complete profile of a patient, and us having the tools to actually stratify and diagnose and treat accordingly using this protocol. So this is where we are, repositories and networks and systems-wide approaches. So you can see that the timelines are fairly stringent, and they are normally about 5 years because the industry doesn't wait much longer. And I think if we streamline with FDA, as I hope will happen as an analysis of COVID itself and the pandemic, that we would be able to move things along for our field in the right way. Now, exciting, and I think Yang Chai knows this, he heard about this from when I talked at the Ectodermal Dysplasia Symposium, and IDCR was also involved in a very exciting initiative. Applications are not that apparent right now because, as I said, craniofacial disorders are mainly developmental, which means in utero strategies are what we need to apply for. But the principle is a good one. The principle is that there would be an adenoviral associated vector platform that would be tried, tested for safety, and well, safety mainly, and FDA approved so that it's ready to go. So if you come in with a specific tissue in mind or specific disease, you would simply add your piece of DNA to the vector platform and then move along with therapy. So this is the phase they're in right now, creating that very platform technology, enhancing vector, looking at, there are not controversies, but there are downsides to putting this in a human, and they are going to work out these side effects, create the right titer or right balance so that it could be used as a therapeutic delivery system. And I think that the diseases that will be targeted are the adult diseases that are genetic, and there are many of them. This is Francis's dream project, and I think before he steps down from his position, he's not retiring from NIH, but he's stepping down as director in a couple of weeks, that this would be already in place. So the idea generation part of it is what I think made a lot of the progress pretty magical for several of these very compound and complex diseases. We have compound and complex diseases that we wrestle with, and I think they're far more challenging just because of the tissues and the scope of the work in hand. Now, very recent came a suggestion from one of the leaders of AMP that we connect all these initiatives by looking at something that would be universal in approach, and that would be, in this case, the systems biology of information. And to really look at information across diseases at individual tissue and cell levels in this very multi-omics viewpoint. So you're looking at genomics and proteomics and metabolomics to create, I think, the algorithms really for successful strategies. And as you know, that the sequelae to our oral health conditions are mostly dealing with inflammation and autoimmunity. So this is really where the concept is currently. And you can see that the scale up is going to happen fairly soon and will be followed by the integrated machine learning data science approaches. So I want to end there. I hope I didn't go over my time limit to ask you to think about these. None of these are prescriptive. None of these, in fact, are critical of the progress made so far, but it gets us to think about the new era we're entering. I ask all of you to put your thoughts there so that we can actually make a difference to the patients that we treat and care for. Thank you very much. And I will leave the PowerPoint for you to study.
Hey, thank you very much, Rena. It was a really great and inspiring talk. I guess I'll hand things over to Matsumi now, and we'll get into the presentations from the individual ITP teams. So thanks again, Rena. It was a great talk. We look forward to the slides and interacting with you on, what the future of regenerative medicine and the doc space might look like.
Great, thank you. Are we ready to move forward with the ITP presentations? If Patrick, are you able to share your screen? Let me know if that doesn't work. Great. If you might be able to put it on the presenter mode. There we go. Bridget, I think we're good to go. Thanks.
Great. So just a few little housekeeping things. So to help keep us on schedule, Kate McMillan is going to help with the timing. So her Zoom video will be a countdown timer. So if you can see Kate or pin her video, you'll be able to watch that countdown timer. So to do that, you go to her name in the participant list, click on more, and then click on pin. Or actually, I might be able to just spotlight. Let me see if this works. Okay, it should be set for everybody, actually. And Kate will play a chime when you have 1 minute remaining, so 8 minutes into the presentation. and for a hard stop at 9 minutes. Then we'll have 5 minutes of discussion, and again, a chime and a hard stop. And then we'll have 1 minute for the evaluators to complete their evaluation while we transition. So if you're an evaluator, you should have already received the pre-reads for the meeting. And this morning, about 15 minutes before the meeting started, you should have received a Qualtrics link from Matsumi with the evaluation form. If you didn't get that, please let Matsumi know. And I think that is about it.
Okay. All right. Well, thanks for that background information. And I think we'll go ahead and launch right into our presentations. We're perfectly on time. So I'm going to introduce the ITP project, a device to preserve adipose tissue grafts for soft tissue reconstruction. And Peter Rubin and his team will give their presentation. So Dr. Rubin.
Hi, thanks very much. It's a real privilege to provide an update to the consortium and the leadership on behalf of myself and our team, Albert Donnenberg, Asima Jaz, and Amy Wiley. Next slide, please. I think someone will need to advance for me. Thank you. So this project has been going very, very well, and I am not going to present everything. Of course, we presented a lot of data on this project. In a nutshell, the goal of this project is to develop a commercializable device that can store adipose tissue for autologous fat grafting on site at clinics and hospitals throughout the nation. And many in the craniofacial space understand that autologous fat grafting has really become an important therapy for cancer reconstruction, reconstruction of congenital anomalies, and trauma reconstruction, in addition to having aesthetic applications. And many patients, in fact, most patients are going to benefit from a second procedure for fat grafting because of the resorption of fat that occurs in all patients during the initial healing and is variable in its magnitude. So this device that we conceived of and we've been working on is a closed system cryo chamber that is single use, it's inexpensive, it works seamlessly with common fat grafting equipment, and will be intuitive to surgeons who use this technique. Moreover, the cryoprotectant agents are very simple. They're simply DMSO and human serum albumin, agents that are commonly in use for cryopreservation and well accepted by the FDA. So please bear in mind that this is a first-in-class device. And on the surface, it seems quite simple. Well, it's just a storage container and a process. But this whole process of working out the science and getting this through the FDA is anything but simple, and we've been making tremendous progress. Next slide, please. During prior updates, I have presented numerous experiments that we have done showing that we can get very effective cryopreservation and viability out to 11 months. I know we can go further, but that's the time period that we had longest that we have assessed so far. You can see in this live dead assay that the viability is almost identical to freshly preserved fat graft tissue. And we have done many experiments in our nude mouse model showing that we get effective healing with favorable architecture of the fat tissue. So I'll just mention this, but I'm not going to go into detail on the science right now because that's been presented many times. And where we are now focusing a lot of our attention is in the regulatory and manufacturing aspects. Next slide, please. In the regulatory side, we believe very firmly that we can get this through in a 510K pathway, and I'll talk more about that in a few minutes. The fat tissue itself does not need to go into liquid nitrogen. It goes into a -80 freezer, which is a lower barrier to entry to have this stored on site in clinics. We believe that at the highest regulatory bar, this will be a 361 product, but we're still working with the FDA on the issue of having this be under the same surgical procedure exemption in 21 CFR 1271. We have had amazing support from the consortium cores, including Kay Fuller's regulatory expertise, the avenues for customer discovery and marketing, and the cell and tissue core at the University of Pittsburgh. We also have been fortunate to have an expert outside regulatory consultant, Richard McFarland, who was a CBER director for 17 years and is now working at Dean Kamen's Institute in New Hampshire. And he wrote many of the guidance documents on the regs regarding 1271. So we had an initial Q-sub meeting. We just had a second Q-sub meeting. I'll get into the details in a few minutes. Next slide, please. Before I talk about that, because I want to talk about that with a few illustrations in front of me, we also have a signed letter of intent with MTF, which is one of the largest commercial tissue banks in the world. They're about $400 to $500 million a year in revenue. They're a nonprofit organization, and MTF sells a lot of acellular dermal matrix and a lot of bone allograft products for reconstructive surgery. And we have not only our own customer discovery data, but at our annual American Society of Plastic Surgeons meeting, they also conducted customer discovery and found a very favorable attitudes by surgeons for having a low-cost method of storing fat for grafting on site. Next slide, please. So let me talk about the manufacturing in the relation to the regulatory components. Our initial design had a filter on one side of the device so that when washing steps were done, you could easily drain the fluid. In our first Q-sub meeting with the FDA, the filter was seen as a big problem because of the potential for this device to be used as a point of care cell processing device, even though that is not in any way the intention of this. The concern voiced by the FDA was that if this was used improperly, that tissue passed through the filter could create a cellular product or something that could be marketed as a cellular product. We are respectful of those concerns. And we actually removed the filter, which is not going to be mission critical for this. We had a second Q-sub this week that was very successful with the FDA. They were very supportive, and they are asking us to move forward with a de novo 510K de novo application. And that actually will give a big competitive advantage to the group. Next slide, please. So our plans for the coming year are going to involve working on the 510K de novo application. We're already dug into that. We're going to have another Q-Sub meeting just to discuss details. From the manufacturing side, we are having our hard tooling, permanent tooling produced. We still have soft tooling from the initial prototypes, and we're having 50 units produced for biologic testing that we're doing in our laboratory to well, we've already optimized the cryoprotectant ratio, but we're going to look at tolerance for using this in patients. I'll pause there for questions. And again, I thank the consortium for your support of this project.
Okay, thank you very much, Dr. Rubin, for the overview on your project and the progress that's been made. I think, summing up some of the key points, you've shown this effective cryopreservation and viability
Yes.
for over 11 months. And much of your focus is on the regulatory and manufacturing aspects and working towards this 510K pathway. So in terms of those regulatory components, I do want to open this up for some discussion by those in that area, because we do see that your project it's very well developed and getting very close to the clinic's door if indeed it goes through this 510K pathway. You recently had that Q-Sub meeting on December 1st. And to see if there are other questions from the regulatory groups at this point.
Yeah, I'd be delighted to answer any questions.
While people are waiting for questions, and you can put them into the chat as well, I'm just curious, as you look at this this collaboration with MTF, and they have this really great track record of working on cell transplantations, materials, allograft materials, those sorts of things. How do you see this over the next year of your project in that association with MTF coming together if everything falls into place?
That's a great question. It's been a really solid collaboration. We've been working with their technical and marketing teams. They believe and they understand that this is going to be a first in class to market. And I anticipate that if we are successful in moving this through the regulatory pathway and continuing the development, that they will enter into either a license or an acquisition of the technology.
Okay. All right. We have two questions here. One of them I see from Lillian Shum at NIDCR. What is your timeline to the 510K? And then also I see Kay Fuller has her hand up.
Yeah. So, one of the reasons we're doing a de novo is because the predicates, there is no single predicate that the FDA will accept for this, and they don't like split predicates. Interestingly, the de novo will actually give us a competitive advantage. And the FDA is very interested, as you can imagine, with the intended use of the product. Therefore, we are designing testing relating to fat viability that needs to be done in a laboratory like ours and will provide good data. We're already doing that. We already had that in our work plan, but this may add a little bit of time. I anticipate that that by next fall, we will be able to, so late in fourth quarter of 22, we should be able to submit that application. Remember, this is a first in class. Please also remember there's a lot going on with the FDA right now and these types of devices. They've shifted control of these devices from CDRH to CBER. And remember, the FDA just finished a 3-year discretionary enforcement period. So this is very interesting times, but we have a good relationship with the FDA. Okay. Impact of work on de novo pathway, I just mentioned that. I don't think it's going to affect that a lot. and then 11 months of equivalency. How long do you, when you demonstrate viability to maximize clinical market adoption? I think we're about there. Most of these repeat injections are within 3 to 6 months. So I think we already have enough there.
All right, and just very quickly, Kay Fuller has had her hand up. So Kay,
Hi.
did you want to quickly ask your question? I know we're almost out of time.
Question and possibly a comment. Thank you, Dr. Rubin. Really good progress. Just to clarify, the de novo pathway is not a 510K pathway, and the information you submit under a de novo is substantially more burdensome, if you will. And so I just want to make sure you're planning for the additional info that would go into a de novo application to eventually get potentially a new 3-letter code to represent your first-in-class product. And then it would become the first, it would be treated as the initial quote product that's regulated to become a predicate in the future for further products. And I just want to make sure you understand it's not a 510K, and have you extended your timeline? Typically, a de novo application takes more time to prepare all the data.
Yeah, no, I thank you. I appreciate that clarification. And
We'll have to be very brief because we need people to have time to put in their comments to do the voting. So very quickly.
I'll just make a concluding remark. Apologies for not further clarifying that as eloquently as you did, Kay, but yes. And we will be happy to be the ones to follow and have that competitive market advantage.
Thank you.
Okay, so thank you. And so now you all have the links to go ahead and post your scores, and then we'll move over to the next presentation.
Bridget, you want me to wait another 30 seconds or so?
Sure.
Okay. I can hear typing, so I assume somebody's entering comments. So why don't we move to the next presentation? So the next ITP presenting is from CDOCTR. The title of the technology is a novel drug candidate for therapy of arthritis, or the TMJ. It's going to be presented by the lead PI, Denis Evsenko, who's Director of Skeletal Regeneration and Vice Chair of Research at USC. And this project is currently in stage 2 with CDOCTR, and we'll be discussing this project for transition to stage 3. Denis, please take it away.
Thank you, Dr. Lutz. Can you hear me okay?
Yes.
Okay, perfect. Well, thank you for this opportunity. And this is a multi-investigator team. You can see people involved in it. So we have experts from several departments, including Dr. Chai's group, who helped us to develop the large animal model for this study. Please, can you advance the slide? Yeah, I'll just walk you through this target product profile, because this technology has actually left the research lab now, and it's entering a late preclinical development and IND enabling studies. So the product that we have developed is a small molecule, injectable small molecule. It's a drug that actually targets a GP130 receptor, a common co-receptor for IL-6 family of cytokines. And the principle of this technology is based on a unique modular inhibition of signaling residues within GP130 receptor that actually shifts the balance within this signaling pathway from destructive outcomes towards regenerative outcomes. And in this case, we are kind of harnessing the regenerative power of inflammation. So the main indication for this therapy is osteoarthritis of the temporomandibular joint. And we are specifically targeting primary, not second, primary osteoarthritis and mild to moderate cases, painful cases. efficacy demonstrated today is based primarily on the large animal studies in pig that we have completed now, and I'll mention our outcome of our conversations with the FDA. So at the moment, we do not expect any additional efficacy studies, so it has been all completed. In terms of the safety studies, the drug has been tested in 2 large animal models now in the different joints, and showed pretty good safety, local safety profile. Systemic toxicology and safety was done in rodents so far. All the studies were not GLP yet. And the regulatory strategy is pretty straightforward for small molecules. On the business side, so this is a completely new class of drugs. It's a new entity. It's very well patented, and the technology has been licensed to a startup company, Carthronics, which is now co-developing it for several indications, arthritis indications. And the plan right now is based on the multiple failures in this field. As you know, osteoarthritis is a very difficult disease to treat, and multiple companies, unfortunately, has failed their clinical trials, stages from 1 to 3. So industry is very resistant to investing in this space. And we expect that we will do the phase 1/2A clinical trial for this technology using startup funds and also using grants, non-dilutive funding. After that, we expect that strategic partners, mainly pharma companies, and we have 2 of them already waiting, if the 2A results in humans are promising, we expect that strategic partner will take this technology forward. And market analysis has been already performed by several groups, some of which were done through DOCTRC. And obviously, there's no disease-modifying drugs for this disease, so there's no really competition of any kind. Well, I think this is just a very quick update. It's not really the data, but just to show you what we have done on the efficacy side. The model that we developed together with Dr. Chai's lab, it's the destabilization model where we remove a partial fragment of the temporomandibular joint disc, TMJ disc, to destabilize the joint. And we have demonstrated now that by 1 month, these animals develop osteoarthritis. It's very similar to osteoarthritis in other locations, for example, in the knee, where meniscus is removed. Yes, it's post-traumatic. There's some limitations, but this is one of the best technologies we can have so far. And then when the disease is established, the drug is injected into the joint. We have conducted 2 injections, and you can see the timeline in pigs, and results were very encouraging, and we have observed significant changes in both cartilaginous portion of the condyle as well as the bone. And this is all quantifiable and pretty significant. And it also resonates with very strong data with other animal models that we have conducted in a non-craniofacial space. So with that, we wanted to move forward to the pre-IND meeting straight, because I think for small molecules, we have some advantages compared to cell therapy or devices with cells. The path for small molecules is very well defined. So we moved straight to the pre-IND meeting. And I think we can move to the next slide. And it's been conducted a few months ago. So what we proposed to the FDA, first we presented all our efficacy data. We presented manufacturing plan, and we also presented a strategy for clinical phase 1/2A trial for patients. So it's, for those of you who are not familiar, 1/2A is a design, it's actually acceptable design, often used for local indications such as injections. So it's safety combined with preliminary efficacy. So it's actually a limited number of patients. And the idea of those trials is actually to define if the drug has efficacy. And you will never reach significance in most cases. Actually, you cannot do that, but you will determine the size of effect, especially for pain, and maybe potentially some structural outcomes. I have shared the results of this meeting with the CDoctor, but this is a key outcome. So we have no additional efficacy studies requested, and it was a pretty standard GLP package that the FDA wanted to see, so which can be completed quickly. So I think next slide will be the last slide, I believe. Yeah, and this is the business strategy that I already addressed. So next slide. Manufacturing also, FDA has no questions. It's a pretty standard process. Next slide. Yeah, and this is, thank you. I think, oh, can you go one back? Yeah, so this is basically a summary of the GLP studies proposed by the FDA. So this is all we need to do to actually put it in humans. And this is a very, very good, package that raises no concerns, but it's expensive. So I just stop here, and I'm happy to answer any questions, because you can see that all we need to do is just to do some safety and toxicology studies in GLP under GMP standards. Right.
Thank you, Dennis. Please either enter your questions into the chat or raise your hand. Any questions? Dennis, while we're waiting, I have one question about what your strategy is for balancing fundraising for the toxicology studies between multiple indications. So as you mentioned, Carthronics is licensed technology, maybe other indications that are being viewed by that company. Are there business considerations in terms of how Carthronics is prioritizing an investment in advancing TMJ versus some others?
Right, this is a very good question. I think, as you know, usually FDA package consists on two portions. One is local toxicology, and then this one is highly specific to TMJ indication in this case. So FDA wanted us to do 2 models, mammalian species, and this is also a systemic portion of safety and toxicology studies. So Carthronics can, and systemic indications, systemic portion is actually the same for all indications. When you come with a new drug entity, FDA wants you to do things like cardiovascular safety to make sure there's no heart arrest, there's no things, even if it's injected in the joint, it's still a possibility, some other things. So the company is willing to match funds to cover completely all systemic toxicology studies. So you can see it outlined in blue color here, because that's essentially, and this is a table that lists all of those.
Great, thank you. So there's a question in the chat about toxicity, and if the systemic data looks good, why are you injecting into the joint? Well, this is a, yeah, it's hard to answer that in one sentence, but first of all, if you know, this is a pleiotropic receptor, targeting it systemically may have some you know, things that we don't want to see. Plus, the drug has low solubility, and we have conducted a lot of studies, pharmacokinetic studies, showing that a single injection can last for up to 3 months above the IC50 level in the joint. So I think there's a lot of considerations related to pharmacokinetics that injecting it into the diseased joint actually achieves much more targeted outcomes with much less potential side effects. Although we did not see any major problems with systemic toxicology so far, but it was done non-GLP. We just think it's just much more efficacious to inject it in the joint. Great, thank you. So there's another question about the activity of the therapy relative to the stage of the disease, and it commercially may be more of a market for late stage OI versus early stage.
Well, we have seen very strong pro-regenerative effects. In fact, in the study that will be published soon, this drug was able to regenerate the entire structure, you know, that we, you know, will report soon. It does have potential for late stages, but we are very conservative. We think that applying it as a joint preservation strategy will be the most efficacious. Plus, our studies have shown very strong effect on pain in addition to structure. In other models, unfortunately for TMJ models, there's not a lot we can do about pain assessment, but The other models have very strong analysis of pain, and the drug has very strong effect on pain, which we believe will be also quite helpful in patients where we just heard a topic on opioids, even in the joint where the disease is not progressed that far, but it's a very strong pain syndrome. It can be very efficacious as an alternative to other analgesics. So I think that's our consideration right now. So we don't want to target very late stages because it's not a magical bullet. It's not going to regenerate the entire joint, but it can definitely have very strong joint preservation effect and potentially some mild regenerative outcomes.
Great. And then another question is how you'll be assessing activity in a phase 1 trial.
Unfortunately, we don't have a lot of biomarkers in this space, and Nancy Lane is a world expert on trials in osteoarthritis. So most of it is patient-reported outcomes. It's based on pain and function reported by the patients, but we will also do some other things such as MRI and some structural outcomes. But the main group of activity outcomes will be based on patient-reported timeline.
In the last few seconds here before we go to the reporting, is there how do you identify the ideal patient for this?
Well, I think we will be very strict with our exclusion criteria. We definitely want to remove people with primary, with secondary TMJ problems. We want to exclude people with neuropathic pain, and I think just focus on primary age-related osteoarthritis of the temporomandibular joint. That's the strategy. So we'll also remove people with autoimmune component.
Okay, thank you. I think we'll have to stop there to allow time for entering your scores. So please go to the sheet and complete those. Thank you. Thank you, Dennis.
Thank you.
We move on to the next presentation.
Nancy, will you be introducing the project?
Can you hear me? Hello?
Yes, we hear you.
Okay, great. Yeah, this next project is going to be entitled Hydronovo. It's regenerating the salivary gland using an injectable hydrogel by investigators Knox, Bailey, and Alsberg and Norhart. And I'll be the discussion moderator, but please go ahead now and start the presentation, which I believe is going to be done by Chelsea. Please, Chelsea.
Thanks, Nancy. Yes, my name is Chelsea Bainey, and I will be presenting this project. But I do want to acknowledge that my co-investigator, Sarah Knox, is on the phone or on the presentation today. She's actually been out on medical leave, but was able to join today despite being in the midst of radiation therapy herself. And really driving home, it's because of the excitement and commitment to this project, which we are very positive about. Next slide, please. So what is our project? Hydronovo Saviginate is an injectable hydrogel that stimulates the regeneration of damaged saliva glands in xerostomia patients for effective and long-lasting relief of dry mouth. And what's important here is we aim to be the only truly regenerative therapy on the market. So we're addressing the unmet clinical need of xerostomia, and specifically our target patient population for the first iteration of this product is post-radiation therapy xerostomia. In later iterations of the product, we look to target other reasons for xerostomia, including Sjogren's syndrome and also drug-induced xerostomia, but we wanted to find a very narrow patient population for our initial clinical trial. So we have been able to receive outside regulatory consultant approval of or advisement of the 502(b)(2) abbreviated pathway. This is a pathway that allows us to rely on the safety and toxicity data of our primary API, which is the drug cevimeline. So cevimeline has been on the market for a number of years and has a very broad and well-established safety and toxicity profile. So we will be able to rely on that data for much of our safety and toxicity. And then it's still a combination device where the drug cevimeline is encapsulated in an alginate hydrogel, and it's delivered directly to the salivary gland. And the alginate hydrogel is going to be viewed based on a predicate, as a device rather than a biologic. So some high-level business strategy here. Our product, we expect that it will be fully reimbursed under the CPT code listed here. We are planning 4 treatments to be used in post-radiation therapy at $1,250 per treatment, and we plan to sell directly to healthcare professionals. We too have benefited from the market analysis, both done by Foresight Technology and also in partnership with the UCSF innovation group at UCSF as part of their tech transfer office. Next slide. So today, last presentation, we really focused on some of the outstanding mechanism of action data that we've developed in the mouse. And we didn't have time to present that here, but I just wanted to remind people from our last presentation that we were able to show that we really have managed to rescue lack of saliva production in mice that have received radiation up to 90 days post-treatment using this therapy. And we've been able to show that we're able to rescue this by a few mechanisms. One, that we reduce cell-mediated death or tunnel-positive death in the salivary gland cells. We actually can induce proliferation of the SOX2 positive stem cells and induce a pro-regenerative phenotype in the salivary gland. And so these really strong mechanism of action data from the mouse are going to support us in our FDA pre-IND application. But I know that the group is really focused on our large animal study. We've developed a large animal dog study that we've been using. And at our last update, we were able to receive bridge funding to have 6 additional dogs. So at the last time we spoke, we were still in the process of setting up the subawards. We now have all 6 dogs in-house and on study right now. You can see the timeline of where these animals are. We need to stagger the animals due to their radiation schedule is pretty intensive, but all 6 animals are in-house and the study completion data is listed. The goal of this was really to get us up between 5 and 7 animals per group. You can see that our Saviginate product is trending much more regenerative than the drug alone, the API alone, and both control groups, either the carrier alone or the control, stay under baseline levels of saliva production. Importantly, we've also seen no adverse effect or severe adverse effect in any dogs or any mice for that matter. And we've completed blinded histology that shows improved tissue morphology. We've shown that previously. One of the things that we did differently in these 6 animals is we're taking a larger biopsy sample so that we can have increased histological scoring. Next slide, please. We've also been working very closely with a number of folks to develop both our pre-IND pathway. At the last update, we discussed how CDOCTR was working to get a new regulatory consultant. That person has now been onboarded. We've had our initial meeting with them, and that's where we have confirmed the 505B accelerated pathway. We have funding to continue working with him on the gap analysis. And we also, I already mentioned that we have no AEs or SAEs. The safety and toxicity will be based on the published API of the reference drug, which is cevimeline. And we have identified the clinical national PI, Sue Yum, who is very excited and anxious to work with us. She is actually in the process of running multiple clinical trials for radiation-induced xerostomia as we speak. She's helped us put together this initial design and has all of the outcome measures standard in her group. Next slide, please. I mentioned that we've been working with the UCSF Innovation Ventures Office. They've been helping us with the commercialization, TAM, SAM, and SOM that is shown here. They've also been working with us on pitch decks and identifying business development opportunities. We've continued due diligence with PharmaCap. We've had almost monthly meetings with them and they maintain interest. And we filed our international patent in 8 major markets. Next slide. The manufacturing, we've had ongoing meetings with both Mike Jameson and Jeff Seifert to ensure that we're addressing manufacturing, packaging, and biocompatibility. We've identified our GMP contract facility, KP Pharmaceuticals, and signed an NDA. We're working with them because they assist and specialize in small to mid volume. We're also working in discussions with US-based API manufacturers and developing all of the manufacturing process instructions. This is an outline of our flow here. Next slide. Okay, so finishing up, this is based, the key milestones here are based only on Q1 2022 because we're in the process of trying to get phase 3. So we have funding through the end of February. And in this time, we will be able to finish the analysis of these 6 animals. We will be working extensively with the regulatory professionals to help us on our gap analysis and prepare for our pre-IND meeting. And then we have the following updates to the manufacturing. We will basically be GMP transition ready and pilot build ready at the end of February. And phase 3 funding would allow us to move into the biocompatibility, ISO, QC, and packaging development. I think we can take questions.
Okay, thank you. Yeah, thank you, Chelsea, that was wonderful. And let's see if we get some questions coming in here. Anybody just take a minute and write everything down. Hopefully my connection is working. Okay. Here's a nice comment. Excellent presentation. Did the FDA okay the canine model to support the IND? Do you have any information on that, Chelsea?
No, we don't have that. I think we will, actually the plan is to propose where we are with the canine and project it as the IND enabling animal. There is no predicate really, so we anticipate that with the data and the development that we should be good to go. We've been working with Mike Jameson on this and we really believe that we have enough safety and efficacy data to move forward into this pre-IND meeting.
Okay. And then another question that's come on, any possibility to move to less than 4 injections in the treatment regimen? Do you think that there's ability to modify that and have efficacy?
Absolutely. I think, though, we probably won't know that until we actually complete the clinical trial on efficacy, versus just because I think that will be important there. And so I think we need to work with our team to identify what will be the initial efficacy phase 1/2 clinical trial on that.
Great. Okay. And does the gel provide a sustained release? Is the potential advantage over the current use of this API?
Correct. It's basically the hydrogel is providing a sustained release. We've shown the hydrogel dissolves in less than 30 days in the dog study based on ultrasound. So we're still able to use it as a drug, not as it's not considered an implantable drug at that point, but the advantage is to give it locally and in a sustained manner. Thank you, Dr. Mooney.
Okay. And actually, I've got a question. The preclinical tox that was done on cerebrolene for the current indication, what animals were those done in? Have you reviewed that?
Oh, of course. Yeah, it's actually been done in everything from small animals. They have toxicity in small animals and large animals, including dog, as a systemic delivery.
Okay. And did they find the dosing, were they able to get to the point that they found toxicity?
They have a very high,
I think it's NOOEL or something.
They have a very high range of toxicity, which is actually one of the reasons we chose it over other muscarinic agonists. So they use it at a very minimal dose relative to where the toxicity was. But the toxicity range is published in their IND application. So we're able to rely on that.
Great. Okay. All right. Well, I think that's pretty much all the questions. So I want to thank you for your presentation and let people write down some feedback. And good. And congratulations. Thank you.
Thank you, Nancy.
Okay, we're now ready to move on to the next presentation. And the PI is Kevin Healy. And
wait, we have a break, I believe.
Yeah, I was expecting a break.
We have a break in for 15 minutes.
I got to know.
Sorry about that. That was my fault. Sorry. Got a little ambitious.
Okay, so we have a break.
Yep, we have our break. So we'll start right at 12:45 Eastern, 9:45 Pacific. Is that right?
Yeah.
Okay, great. Thanks.
Okay, I think we're ready to start. Kevin, you back?
Yes, can you hear me?
Yeah, perfect. Okay, hi everyone. We'll start with our next presentation, bio-inspired hydrogels for craniofacial muscle regeneration. The PI is Kevin Haley from UC Berkeley. Kevin.
Good morning, good afternoon. Can you advance the slide, please?
We put the presentation in the, yeah, okay, great, thanks.
Great, this is our team. Thanks, John, for the introduction. Just pointing out both myself and Anouk Kellars, the lead postdoc, are at Berkeley, George Christ at Virginia, Jason Pomerantz at UCSF, and Glenn Presswick is at University of Utah and also Washington State University. Please move forward. So our TBP is a hyaluronic acid-based hydrogel for volumetric muscle loss. We've talked about this before, so I'll just go over this quickly. Our indication is VML injury resulting from permanent cosmetic and functional deficits. And our first IFU will be for muscle repair after cancer resection surgery. The product is a hyaluronic acid microporous scaffold. We'll talk about that a little bit later. And our regulatory strategy is medical device with a plan for a startup in Q4 2022. Next slide, please. So key projects, only one slide on the technical. So I'll just read through sort of our major ongoing efforts, which is scale up and manufacturing, which I'll address in a moment. It has been completed, so a 10x batch, that's really important for us. The microporous scaffold fabrication is ongoing, and that's being done with a company in Oregon. And then histological analysis based on our muscle regeneration experiments are ongoing, and analytical methods development related to manufacturing commercialization, are ongoing. So the scientific update, I thought it would be important to address the histology. Questions were asked in the last retreat, or I should say the June breakout retreat, where we were asked to show two things, possibly regenerating cells in the area of the injured tissue. So that's what you see in this PAC7 staining of the satellite cells that are co-stained with Ki67. And then looking at neuromuscular junction that might be occurring with the implant in the implanted area. And that's shown by the staining on the right at 8 and 16 weeks. So we've completed this 16-week study with the gel. We also have in the TA model another study that was long-term that's been completed, and both sort of point to the same conclusion, which is reduced fibrosis and regeneration. And in the TA model, we're able to measure functional force regeneration with nearly 100% return on the deficit in that particular model. George, and on the left, the slides were from Jason Pomerantz's group. And then on the right, George Chris is now looking at the injured area in the masseter with a program he calls SMASH. So this is a sort of semi-automated analysis of basically the histology, so that we will be able to get a fiber cross-sectional area and other fiber coordinates, if you will, in this injured area. You're looking at an uninjured area, and that's an ongoing area of effort for us. Next slide, please. So the project updates for regulatory and commercialization have this color-coded. Green means completed. So the tide mark and the gradient between green and red means ongoing. So, you know, it should say microporous scaffold. Sorry, that's a typo. So we have the TPP indication for use. We've gone through this. Maybe pointing out that we've been in working with Patsy Trissler as our FDA consultant and Mike Jameson, and the plan is for an information meeting with the FDA in Q2 of 2022. We've gone through GMP Light pilot scale up of the macromers needed for our system. And now we're starting to move into the lyophilization, sterilization, and a large part of our ongoing work will be analytical methods development for this, what we call this volume matrix is the scaffold. And finally, probably later this year, stability and degradation of the scaffold. Next slide, please. So commercialization, as I just mentioned, Richmond Chemical is doing our CMO scale up. So we've completed a 10x batch, which is basically going to produce enough gel for all the studies we've proposed in the next year. So that's a good sign. We have commercial grade components that are for the acrylated hyaluronic acid, the peptide acrylated hyaluronic acid, the thiolated heparin, and then adhesion and cross-linking peptides. And now we're moving into freeze drying with Oregon freeze dry of basically taking the hydrogel and creating the scaffold. And we're in the process of getting all the materials that are made commercially at Richmond to Oregon freeze dry. We've transferred the SOPs. Really, we're just about to initiate that whole project. We had a little bit of a delay. They've had a change in personnel and also moved to a new facility. So that caused a delay in getting that part of the project started. And then we haven't yet initiated work with Nova Sterilis as a supercritical CO2 sterilization or selection for our mode of sterilization. But that will start in the next quarter. Next slide, please. And then on manufacturing, this is just eye candy showing you, this is what we have Richmond making for us, this acrylated hyaluronic acid, the peptide modified acrylated hyaluronic acid. They've been working on a great deal of analytical methods for us. So I'm just showing you a simple NMR, but GPC, HPLC, and GCMS, these are all techniques that we're going to be using to validate and characterize the CMO produced material. And then the TFF, I just want to show this because the funding for that little instrument came through CDoctor, and now that's at Richmond where they're doing small batches and using the TFF for filtration and purification of the macromers. So we're in the process of creating this, we're calling it GMP Light batch, and we'll report on that at the next retreat. Next slide, please. And then the plans for the next 6 months are mostly on the technical side are just transferring what we've been doing with the gel form to the TPP is basically creating the scaffold, characterizing it, and then starting our implantation studies with the scaffold. Chris is developing, and this has been requested by this group in the past, a masseter force measurement apparatus, and he's on the call. He can comment on that in the Q&A. And it's SMASH, as I mentioned, histological analysis. Both of those are being developed. And then I mentioned regulatory. We're going to do the pre-submission information meeting in hopefully Q2. Ooh, that was loud. and then I'll just say that our IP position, it says ongoing patent application, but that I didn't point out, there's already an issued patent on the material, basically compositional matter for the gel material. And then this patent that's being prosecuted is for indication of use in the craniofacial area and muscle regeneration in general. Thank you.
Thank you, Kevin, for that great update. Glad to see there's so much progress made. So people, please post your questions in the chat or raise your hand, and I'll start. Maybe George can share with us, what criteria will you use to evaluate the physiological function of the regenerated muscle.
I assume George was on the call, but I don't know for sure. George, are you there? Okay, so I will, it is in, let's see, it's in the report, I believe. No, I'm just looking at the report, which had a little more data than this. It's actually a similar type of physical force measurement on a force measurement, I'll just say force measurement apparatus, where the probe, if you will, is hooked into the animal's jaw. And that's about all I can say on it, only because it's just under development. I think George has got 1 or 2 animals where he's tried this on. So it's early on, but I want to point that out, that's I think an area and a question that we've been able to handle in the TA model, where we can easily measure the force. Yeah.
Okay, so the next question is whether your formula contains heparin.
I'm sorry, Jan, one more time.
So question from Kay Fuller, whether your formulation includes a heparin element.
Yeah, it does. A very small, I think this came up previously, a very small mass of heparin is thiolated. And that means has a bunch of SH groups that basically entrain it and keep it locked into the hydrogel. So it doesn't distribute. And that formulation we have shown doesn't have any, well, we haven't shown, we anticipate not having any effect on anticoagulation, but that's a test that we're planning on doing.
Question from David Mooney. HA product has been shown to the breakdown have some biological activity. Do you know whether this play a role in the regeneration in your application and the potential impact on regulatory paths if that occurs?
Yeah, thanks for the question, Tim. We don't know about the regulatory path. I think that's part of the information meeting. So we'll get more guidance when we have that meeting. But we're going to argue no. Just to stick on this message, we're going to try to make the same argument that folks make for decellularized matrices and that have got them approved as devices. So the consultant we've hired is one that was introduced to us by Steve Badalak, who many of you might know on the call, that has had his decellularized matrices regulated as devices. So that's the approach there. Regarding the breakdown products, we've done a pretty, I think Dave's referring to that hyaluronic acid could be an angiogenic stimulant. You know, I've gone through the literature, and I think there's definitely a molecular weight dependence on that, and it has to be pretty small. Yep.
Kevin, this is Glenn. Yeah, this is Glenn.
Oh, Glenn.
One of the things that we're not particularly worried about is small fragments of HA, simply because when the fragments are produced by the degradation of the scaffold, those fragments will be either peptide modified or acrylate modified or connected to other fragments, so that they're not going to be freestanding HA fragments by themselves. And we'll be looking through LCMS methods to see what the fragments are when we develop our methods analysis, and what peptides are produced as a result of degradation, either with heat or oxidation of the scaffold.
Okay.
Thanks, Glenn. But one, and to add, I think regarding the question that this activity of HA is molecular weight dependent on small molecules, and I think there is a bit of controversy in the angiogenic effect.
Yeah, everyone, please make sure you enter your evaluation. So we have a couple more questions. Will muscle innervation be a metric for success?
I think we showed the early histology. I think the muscle innervation will judge by the quantitative force measurements, but we will back that up with histology showing that there's clearly innervation in the regenerated muscle.
Are there strategies in place for the commercialization plan and licensing industry partners, etc.?
Yeah, the licensing will go through both UC Berkeley and University of Virginia. And the strategy is to form, as I said, a company in Q4 of 2022 and probably take the path of non-dilutive SBIR and or maybe we'll go in one of the local accelerators like we did with some of our other companies, either the one here at Berkeley, Skydeck, or Y Combinator, one of the ones in the Bay Area.
Yeah, so there, I think that chime means we're out of time. So we'll move on to the next one. And there's still another question from Dave. Kevin, maybe you can just respond in the chat.
Yes, thank you. I'll respond to Dave Cohen in the chat. Thank you. Thanks, everyone.
Okay, so the next session is presented by Michael Longacre from Stanford. Take it away, Mike.
Thanks, Ben. This is a project we've had funding for the past 5 or 6 months. And it's the one-time local injection of verteporfin to promote wound repair via regeneration without scarring. Next slide, please. So the clinical unmet need is cleft lip closure, about 7,000 per year, primary, and that will put it in an orphan drug indication. It's used to prevent scar formation in this population. It's also possible that we could consider cleft lip scar revision in slightly older patients, and that will be determined in discussions with our regulatory consultant. It's an injectable local solution that's applied during the closure. We have demonstrated significant small and large animal models for drug reduction, for scar reduction. Safety is well known. This product has been used for an IV for macular degeneration for over 20 years. We anticipate an orphan indication with a 505(b)(2) repurposed pathway. The technical risk for this has been addressed now in both large and small animal models, and our large animal model data are being prepared. We've had multiple conversations with both strategics and venture firms who have strong interest in this market. And as I said, the initial market is cleft lip closure. The burden of scarring on those children in form or function is well known. Parents will be highly motivated to minimize the scar. And the commercial partners demonstrate the size of the market. Our paper was published and got a large amount of lay press, New York Times and Wall Street Journal, et cetera, in Science. We've subsequently moved on to red Duroc pigs at the dose of 2 milligrams and demonstrated that we also see, and these were very, very tight closures. The wounds were closed by the 2 craniofacial surgeons who will do the cleft lips. The material was mixed in a GMP sterile facility and witnessed and signed as you would for a GLP study. Significantly, we show that the wound breaking strength is normal compared to normal skin. That's important for no scar, and they are significantly less stiff. There's no systemic drug was discovered 1 day later. And there's no histopath toxicity on these pigs. I'll simply say that we're finalizing the manuscript, but there is a population of fibroblasts we've published extensively in this area that is present in verteporfin and unwounded skin, but is not present in PBS. So we're really excited about that. Next slide. We hope to secure a new consultant at RPI, as multiple people have mentioned. That's our consulting group, and Michael Jameson is working on this. We hope when we finish the large animal study that we could begin pre-IND preparations with our regulatory professionals. Cleft lip closure is an orphan indication. This is a repurposed drug. verteporfin is off patent. The pig study was performed, as I said, in a GLP-like manner, including histopath and dosing with no systemic or targeted effects. We'll initiate experiments on non-clinical safety and support of pediatric drug medicines, and that will be in 2 species, but this will be guided by our regulatory consultants. We have a human xenograft model, which we will have data in 2 weeks, but preliminarily, that is human skin on the back of nude mice, which is a model that we've published recently, so we're excited about that. Next slide. We hope to complete our large animal study and write it up for publication. We'd like to engage with our new consultant once that is nailed down, and I believe that is very close to happening. We will have our human xenograft wound data complete by the end of December, and it's possible that we would engage Stanford Alliance for Innovations in Medicine, potentially for an NCE, but we haven't finalized that yet. Manufacturing CMC issues have been addressed as verteporfin is a commercial product and now off patent over the last 20 years. IP has been filed for the indication of scarring, both in the US and now abroad in large markets. No prior art has been discovered, so we're excited about that. And we've had multiple conversations with potential partners to commercialize it. It's also possible in terms of funding for our clinical trial, first in human, we would look to the UH3 clinical trial, and Drs. Wan and Gertner have applied for that. So we will be following that closely. In terms of CDoctor support, Michael Jameson has been invaluable. And I've been following the progress of Drs. Gertner and Wan as they're moving forward with their own projects. Next slide. Milestones, I think technical risk, which is the risk that is, does it work, has now been addressed in purebred red Duroc pigs and mice. I would say 85 to 90+% of the risk of this working in humans is now wrung out. Efficacy data in this animal model strongly supports there will be clinical efficacy. And again, this is by 3 different levels. Tension offloading dressing has been used on over 250,000 patients with 2 randomized controlled trials from a company. Dr. Gertner has published on FAK disintermediation in both mice and pigs. This verteporfin is a YES-associated protein inhibitor, the last step in the bucket brigade of mechanotransduction. And now we have that data coming together in mice and pigs. The human xenograft data, I think, would be the icing on the cake, and we'll have that in the next 3 weeks. So based on 3 levels of evidence, skin tension offloading with a commercial product made by a company, and then FAK and YAP, we strongly suggest this will work in humans and result in a go decision for our group. Next slide. So plans for the next 12 months. We will finalize our single cell RNA-seq and pseudotime analysis. We'll also have the data on the human skin wounded that's been injected with verteporfin. Again, this is a one-time local injection at the time you close it. This is in strong contrast to systemic IV use in tens of thousands of patients for macular degeneration. Regulatory, we want to identify our new consultant, and we should be ready to go on that shortly, beginning when we have all our large animal data collected, and we hope to submit that for publication in the next month. Commercialization, the people we're talking to like the fact we've addressed technical risk in a large animal model. They certainly understand the idea of a primary orphan indication with 85% of drugs on the market going that route. Follow-up markets, no doubt, are huge, but we're going with a pediatric craniofacial indication. Manufacturing, Bausch & Lomb makes Visudyne, so that is not going to be an unknown in terms of going forward. That's already in use for IV use. We look for our IND process to begin, and we'll be working with our regulatory consultant, and Michael Jameson has really been helpful in that with the Gertner and Wan projects at Stanford. I think that's my last slide. Thank you for your attention.
Perfect timing, thank you. Let's take some questions from the audience. While people are waiting, maybe I'll just come up with one. Mike, very impressive data in such a short time. In other models, not in the cleft lip model, have you compared just head-to-head between tension offloading type devices and verteporfin in indications where actually the tension offloading devices could be applicable?
Yes, and that's a good point. So at the skin level, 2 randomized controlled trials have shown that strongly reduces scarring, but not to the level of true regeneration. I do have to say that we have a novel neural network AI program that showed that 300 features of each ECM fiber is normal, that is normal. Biomechanics were restored and secondary elements were there in terms of hair, et cetera. None of those have happened with the tension offloaded device, but still has significant visual analog data. So we're trying to get objective here. And we haven't compared it head-to-head, but this is true regeneration, and that is significant reduction of scarring.
Thank you for the clarification.
Could I ask a question about the potential for a need of follow-up injections locally after the closure? It's sometimes the scar formation, at least from some of the stuff that I've followed from the laser center down at UC Irvine, the Beckman Laser Center. They were finding that the traction forces acted over time to increase the scar. And I'm wondering if one injection is adequate or if you have plans for addressing that.
So excellent question. Certainly very different in terms of laser resurfacing. So we just published a paper in PNAS last month about the mechanofibrotic subpopulations of fibroblasts. So it turns out as they come in on day 4 or 5 in any animal model, including humans, if you can block the activation of that subset of fibroblasts, which would include engrailed, what we talked about in the science paper, that we follow these animals out. In the pigs actually have been followed out for 16 weeks with no erosion of benefit. And if you, in fact, if you look on that slide carefully, and you'll see it in publication, that they continue to outperform in the remodeling phase PBS. So we don't have plans for that right now because we have not seen it necessary, but obviously if it comes up, that would be a very straightforward thing to do in a patient.
Excellent, thanks.
Mike, Will Ginobili has a question. If you could mention briefly the plan on UG3, UX3 clinical grants that has been prepared.
So I'm not an expert on this, Will. Thank you for the question. Doctors Juan and Gertner have applied for that, and that is a mechanism which the NIDCR has been very helpful in guiding us towards. And it's a potential way for us to fund our first in human, but I haven't begun the conversation yet. But it is a very helpful pathway, and NIDCR has been very supportive of Jeff and Derek.
2 questions about the drug. Has Bausch and Lomb agreed to manufacture your indication? That's from Bob Spiro. And then Lillian Shum has a question about how does dosing work for local application injection over incisional?
So 2 good questions. Bob, haven't done it yet, but it's off patent now. And I don't anticipate that being a bar at all for them to be able to sell more product. But we haven't begun those conversations formally, Bob. Lillian, how does dosing work? So dosing was worked out from mouse to pig, and I didn't show you the data on how we got to 2 milligrams, but that is also the best, the dose most optimally giving us regeneration. So it's a local injection. Obviously, when this drug has been approved and given in tens of thousands of patients IV, that is a significant advantage for us. In terms of Pat Olson's question, if I may, not sure yet. We're going to finalize and submit the pig data, including the mouse xenograft data in the next month. And then we will be meeting with our PI to plan for the pre-IND. What makes the clinical application specific? Well, I'm a craniofacial surgeon, and that is near and dear to my heart. essentially, and this is a slightly overstatement, all the burden of those children is in scarring on form and function and growth. So that's why I like to do that. Obviously, there's 100 million incisions a year in the United States, and I'm sure the commercial partners will address that with secondary markets. Any thoughts on traumatic lacerations? Secondary markets, Peter, there are about 20 million of them, but we're going to stick to a very focused crossing of the chasm in cleft. But obviously 100 million other incisions and lacerations per year, US alone. All of those heal with a scar.
Thank you very much, Mike.
Thank you very much, everyone.
Okay, we're ready to move on to the next presentation, stem cell-based therapies for bone regeneration around alien dental implants. And the PI is Tara Aglu from UCLA. Tara, go ahead.
Thank you very much, Dr. Chai. Thank you all for being here today, and so that I can discuss our project together with Dr. Alireza Moshe-Verinia. Next slide, please. Thank you. So our target product profile, actually, I was going to have those come in one by one, so maybe not go yet. Yeah, thank you. Perfect. Okay, great. So this is really targeting peri-implantitis, and those of you who work with implants or have seen patients with implants, you know that this is an infectious disease where there's a major inflammatory process locally around implants that are osseointegrated, and then they lose supporting bone. And nowadays, because implants have been in for many years now, over 30 years, and we have a lot of patients that either have systemic diseases or local problems with wound care and hygiene, that there's a significant problem where up to 56% of all implants have peri-implantitis, which is many, many per year. And so we have 400,000 cases of peri-implantitis, and that's going to continue to increase. Thank you. So our current therapy really is very limited, and it's debridement of the implant or removal, because the major problem with debriding or cleaning up the implant is the lack of ability to regenerate bone. And so even though we can maintain and clean up the area, completely decontaminating the surface is almost impossible. And then the regeneration of bone is very unpredictable. And so the costs are significant, $1,500 to $3,000 per implant. And then as we have more patients with larger reconstructions, that gets even more and more expensive. Next part, thank you. So our product is an osteoconductive light curable adhesive hydrogel with MSCs that has the ability to regenerate bone. And when we look at this, so far we've shown in these meetings the ability for osteogenic differentiation in vitro and bone formation in vivo, both in small animal as well as large animal environment in this inflammatory environment. So we've been working with pigs, and we've looked at not only the efficacy, but then the safety in terms of both local, regional, and systemic inflammation and lack thereof in this pig model. And when we think about the regulatory strategy, that so far we have always really planned to be with CBER, and we were going for an Interact meeting, but the recent discussions for the FDA with Yang Chai's group have kind of made us rethink a little bit, and possibly a potentially more simplistic type of FDA approval. And so that is, as his group is going through that now, and I'm sure we'll hear more about that tomorrow, Yang, and then we will be following sort of the same way that that Dr. Longanker was talking about, following some of the others before, which is really one of my favorite parts of the CDoctor, is because we have so many groups at different stages that we're able to have some be the pioneers in certain areas, so that the rest of us maybe can have a little bit more streamlined and set up approach. If we think about the business strategy, this has been a real struggle for our group, because I know some of you, from what I can tell from either your experience or just how you're speaking about it, you really have in mind whether you want to have a startup company. When I was listening to Kevin Healy and he said, oh, well, we'll do a company just like we have with our others, to our group, that's a little bit more of a daunting task. And so, particularly this has been such an area of agonizing discussion for our group and thinking about it. And we've had some really great seminars in the Innovator seminar series that Bridget and the group have put together to really help us make some of those decisions. And really my experience and expertise as a clinician, as an oral and maxillofacial surgeon, has been working with large companies, and several of them have expressed interest, both the implant companies and the regenerative companies. And we've actually spoken with the CEO of one, Geislich Biomaterials, and the R&D head of Straumann, one of the implant companies, and have had some letters of interest from them as well in potentially licensing the product or acquiring it. And so, again, just really making that final decision on which route to go. So that's where we've been spending a lot of our time thinking about it. Next part, please. So when we look about, this is the clinical problem, and you can see in the radiograph on the top that we have bone loss that's pretty significant around the implants. And when you open it up, it's a real major problem. Either we're going to have to remove those and with a major expense. And you can see how it affects the teeth close by and even the other implant in the vicinity. Next part. And so this is really our product. And the reason that it's so cool to think about this in the dental office is because every dentist is going to have a light curing unit, a handheld one in the office where this can be used, where you would get this, this package and you'd be able to utilize it. It's adhesive to titanium, to bone, to periosteum, to gingival tissues. And then when you put it in, it's even a bit more than a paste. It's more of like a composite material type of consistency. And so it's really easy to inject and mold into the area. And then it's light curable and can bind both to the implant and the bone after the regular debridement is done, as would be done with any normal periimplantitis treatment, but then focusing on the regeneration. Next slide, please. And so some of our key project updates. So because of using this obviously in the mouth, we have to look at the degradation in saliva as compared to PBS. And so this was done, and you can see here that this aspect here is looking at in human saliva. And certainly we don't want the hydrogel to be around indefinitely, because there are a lot of issues with that. With the residue, we want it to go away and we want the new bone to form. And so we don't want it to be super long, but obviously not to be very, very fast as well. So this is our final formulation here. So going out to about 5 weeks, 3 to 5 weeks. And when the bone formation begins and the degradation is very relevant and makes a lot of sense in terms of clinical use and a clinical product. Next slide, please. And then, so really that was our go, no go that I just mentioned. So with the regulatory aspect, this is really where we have been thinking a lot about and looking to other of the C-Doctor groups here locally, because some of the feedback, of course, many of you probably remember when we first started, we were thinking about autologous MSCs because we wanted to get the most robust type of bone formation that could occur. And then of course, the regulatory simplicity would have been much better with allogeneic MSCs. And so our initial both small and large animal studies were done with allogeneic MSCs. And there was no significant difference both in terms of efficacy and safety when we used the allogeneic. We actually had identified some allogeneic MSCs for human studies, but unfortunately those were outside the US. And so we weren't able to really pursue that. And so that again changed things a little bit pretty recently. And so now we are looking to await the final decision from the pre-RFD FDA meeting that I know Yang Chai's group had to look at bone marrow aspirate in terms of utilizing that to even further simplify the regulatory aspects where it doesn't even have to be taken out of the operating room and then go back in. So using the bone marrow aspirate to place at that time. And we've had our initial regulatory proposal and consultation with Biologics Consulting. Next part, please. So these are our go, no go right now to be done very soon. finally, next slide. Go on. Again, I talked about this in terms of the commercialization and what has been done and what is going on now there. And next slide, please, which is the last one. Manufacturing, again, just the identification of all the GMP materials to utilize for this. Next slide, please. And this is really our final, where we are finalizing the bone marrow aspirate studies, awaiting on the pre-RFD, finalizing the commercialization. And one thing that I really would still like from our groups is if we could have a panel discussion with a lot of the other C-Doctor groups that have already done this, so that we could get feedback from you on what the pros and cons and benefits are. And then another thing would be the introduction to the local GMP and GLP facility that we would be working with. So thank you for your attention.
Thank you, Tara, for that great update and questions. So I'll start. Have you looked at the different dosage of bone marrow aspirate in your model, and then try to decide what is the ideal loading dose for bone regeneration?
No, that's what we're doing now. And, and in fact, that's one of the things that, if we're having more of the discussions with other C-Doctor groups, because that was a little bit new to us and wasn't exactly in the plans. I do a bit of bone marrow aspirate use in patients, but for this particular situation, then that is something that we will be doing in the next time, is looking at the dose response in terms of, obviously the lowest amount of dose and the maximized osteoconductive effect with the scaffold. So that's something absolutely that's planned for the next 6 months.
Great. And the next question is whether the light source you're using would damage the MSCs.
Well, it has not in both the autogenous and the allogeneic. So, if that's going to be any different in the bone marrow aspirate, I wouldn't imagine, but certainly that's a great point and something that we need to look at, because, since we did that with our other cells, then as we transition to the bone marrow aspirate, then that's certainly something that we would need to repeat as well.
And Nancy's question is, what is the rationale as to why the MSCs will be effective, and what is their role?
Well, the MSCs, obviously, if we have MSCs that can be differentiated into osteoblasts, the hydrogel has HA microparticles. And so we had shown that when the MSCs were placed with this osteoconductive hydrogel, then they were differentiating more quickly and more robustly, mineralizing and differentiating and forming bone in both in vitro and in vivo situations. So, now with the change of using bone marrow aspirate, it does make a lot more sense, and certainly that's something that's already being used in the clinic. And so I'm very, I think it will simplify things quite a bit. And so I'm looking forward to having that, and maybe Yang, I don't know if that's something you'll update us on tomorrow about how close the FDA is to saying if that's going to be acceptable in terms of your RFT, pre-RFT meeting.
I'll provide an update.
We won't steal your thunder right now, huh?
Question from Darnell. So have you thought about comparing the effectiveness of using bone marrow aspirate versus a more potent MSC population?
Yeah, I mean, Darnell, that's a great question. And, we will be doing that in the comparison, but I think we're kind of limited anyway because of the MSC population in terms of using allogeneic that really still aren't available. I mean, the ones that we were planning to utilize had been in, they're manufactured and through GMP and everything in Germany and are being used in Canada, but couldn't really be used for this purpose in the US right now. And those are already in clinical trials. So certainly those were ones that we had previously identified. And so they will be compared with the bone marrow aspirate, because you're absolutely right. that, we're not obviously looking ahead, we're looking at larger infected defects such as infected fractures and mandibular, both marginal and continuity defects. And so those are going to be even more difficult defect healing and bone regeneration. But for these very, very small defects, we're very optimistic with utilizing the bone marrow aspirate, especially with the results that we've seen that that Yang's group has gotten with bone marrow aspirate.
More questions, whether you have received any requests from FDA for your pre-RFD submission. My understanding, you haven't submitted the
No, because we didn't want to basically repeat the wheel until we knew if this was the strategy with your product, and then we were basically ready to follow suit with that. So we've already met with Biologics Consulting and had them give the initial our initial plan for FDA for the pre-RFD meeting, so that's all. We'll all be ready to go as soon as we have the final decisions.
Okay. Maybe one very quick response. Do you know if age or pre-existing conditions affect MSC-directed bone formation in this model?
So that's a good question. We don't, and certainly, this was a question in terms of what will be the specific indication for this product, because, obviously there are patients that are more prone to peri-implantitis, such as patients who do have medical conditions like diabetes, or smokers who do have, more inflammation, and they're prone to bone loss, and specifically peri-implantitis. And so, we haven't done that in our animal models yet. And so if that's something that, either the group or the FDA would want us to do, then certainly that's something that we would need to do. Absolutely.
Thank you, Tara. If you can just take a look at the questions, there are quite a few more in the chat, and respond to them. And thank you, everyone.
All right, our next presentation is optimization of a novel organic mineral bone adhesive for dental bone grafting. Product name is Tetranite.
Okay, thank you. Grayson and I will be sharing the presentation today. I want to thank everyone for their help and support over the last couple of years. So Grayson, why don't you take it away? Can I have the next slide?
We're not hearing if you're presenting.
Grayson, can you hear us?
Joe, do you want to take over?
Yeah, why don't I take over then? So, as we've said before, the description of what we're doing is we're using tetracalcium phosphate with phosphoserine, and to look at a novel way to maintain or grow a ridge and alveolar bone. And the unmet need is pretty extensive from a clinical point of view, the loss of bone, and then in order to build it back, to use membranes, particulate, tacking screws, etc., makes the whole procedure a little less predictable. So our focus is to look at onlay grafts, horizontal augmentation, so that we can place dental implants. And as I said, the technology really allows us to stray away from the use of, say, block grafts and screws and membranes, and then resorb in a timely manner. We've had a lot of updates from the regulatory point of view. Either Grayson or probably Brian can get into that. But we've been classified as a de novo now. I know that the first presenter, that's Ruben, talked about the de novo pathway. There's been a lot of work on the regulatory side. The IP is pretty strong. There's been IP issued through Stryker with Brian as the inventor, and then additional patents have come forward. And the business model, I think, is pretty sound. And we've been working with the Avenues Group to help look at getting key opinion leaders and anticipate of what the indication will be and user handling, et cetera. So next slide. So we've really gone to the FDA with a couple Q subs, and now there's, again, maybe Brian or Grace will come in with the regulatory issues, but we're going to have two 12-millimeter defects per hemimandible, at least 10 millimeters inter defect space. There'll be 3 cohorts, 3, 6, and 9 months, and will be a total of 24 animals. There's been a lot of work done on the outcomes. Obviously, we've, even in the phase 2 of this trial, of this grant, looked at histology and in measurements. We've validated a CT analysis method, again, to look at ridge preservation compared to the market controls. Compression testing, which is another FDA requirement. And then a lot of work has been done on the PK testing and looking at the metabolites, and that's been validated by an external vendor. So we're waiting again for budget approval to again continue with this path. Next slide.
So from a regulatory update perspective, the big news is that we received confirmation from Dr. William Mizell, who's the head of all product evaluation at CDRH, that indeed the de novo pathway is the most appropriate pathway to assess this device. We currently have a pending pre-submission filing with the FDA, and that's focused on confirming the animal study design, and then also addressing and discussing the specific controls that we would put in place in our ultimate human trial to meet the de novo pathway requirements. Next.
So there's been a lot of work on the commercialization side. Here you can see the final packaging. We've been playing with it and testing it. Can you click through so you see all the, thank you. One more. Go back one. It's a pretty nifty little kit. It has a mixing bowl, the liquid in the syringe, the powder, and the spatula. And it's very user-friendly from the dental point of view. And we're just going to be going through some more validation trials, again, with the Avenues group, looking at shelf, life testing, et cetera. Next slide. Other things that have gone on from the manufacturing side is that we've optimized the granular yield in the manufacturing, which is real important as we scale up. The liquid is being all filled by machines because it needs to be very precise before the packaging. And a bill of materials is being developed again for the manufacturing. It's sort of the SOP or the sort of the recipe for the manufacturing, whether it's in-house or with outside vendors. Next.
So the key, So the key planned activities over the next 6 months are essentially the most critical path is proceeding with the pivotal animal study and enrolling the 3 cohorts that Joe mentioned in terms of the study design. And of course, the material will be assessed in 12-millimeter box-shaped defects to show bone ingrowth based on the parameters previously discussed. And in terms of regulatory update, we will get a readout in early January on the existing pre-sub that's under review. And that will, again, confirm the study design and also explore the special controls that are needed for de novo approval. In terms of commercialization, one of the key elements that we're looking to do is to do additional user handling trials to make sure that the kit, its design, the component parts, and the way that our instructions for use are being developed work from a clinical perspective. We've already had previous sessions of this kind, but we need to go through what's called a summative user handling trial, and that's what is on the plate. In terms of manufacturing, we're actually ahead on this key task, because as you saw, we've already released all the manufacturing documents and approved them internally, and that's all in conformance with our quality management system. And then of course, other key elements for the overall product development so that we can file an IDE with the FDA is to complete biocompatibility testing, sterility validation, shelf life testing, and draft our IDE submission. I think that concludes our presentation. We're happy to take any questions you might have.
Great, thank you. So the first question that we have is, does this material have adhesive properties? If it does, what is the strength?
It is adhesive even in a wet field. Brian, you may want to chime in on this a little bit more.
Yeah, the underlying technology itself is adhesive. That's really the unique property. We currently have a clinical trial ongoing to stabilize and adhere dental implants to bone. So it's adhesive to both metal and bone. In this application, though, we're looking at larger volumes of the material, more porous material. And so you saw an image before of these preformed tetranite granules that are porous. And then we bind those together with the self-adhesive form of the material that sets up inside the body. And so it's flowable and cohesive, but also adhesive to bone. The strength can vary, but it's very robust and eliminates the need for membranes and tacks. We look at shear strength and compression strength, all different modes of bond strength of the material to bone.
And as it sets, you're able to shape it. And once it sets, you're able to adjust it with hand chisel. I mean, there's many different instruments that clinicians use to do that.
All right. What do you think led to or what do you think the key factors are that led to the de novo pathway?
Part of that is, so dental bone graft materials were declassified from a class 3 to a class 2 device status. And so therefore, dental bone grafting is a class 2 category. The concerns were initially from the FDA were that it's a novel material. However, as we continue to have pre-submission meetings with the FDA, they confirmed that indeed phosphoserine is not a novel molecular entity. And so therefore we could fit into that de novo classification. Does that answer your question?
Yes, yeah, that's great. I'm going to go ahead and ask it. Oh, here we have one. Can you comment on the desirable porosity and degradation rate?
Yeah. So we've been, I think, over the past several years with the center, we've gone from lower porosity to a higher porosity. So we're probably at about 35%, but also the granules have become porous also. So I think the key thing is to get vascularity ingrowth in that. So in our prior preclinical study funded by the center, we had very good optimization of the porosity. We've done a little tweaking of it, but we had good bone ingrowth and replacement in a clinically relevant manner, given sort of the proportionality of the canine to the human.
So I know there's been a challenge with the FDA in terms of their recommended animal model and how aggressive that model was or should be. Can you comment on that interaction and what you think the future pathway is in that interaction?
So I think the group has had several challenges with the FDA, I think both in this study and the stabilization product in terms of preclinical guidance. I'm not going to comment on the stabilization, preclinical studies that were done. Here we did meet with them. There were clear focuses by the group in terms of negative controls. They weren't so much worried about the market controls. I think those were pretty much what we had. And again, to satisfy their vision, we went with a larger defect and more defects per animal, which really caused some issues in terms of healing. And we've gone back and rectified those, I think, in a very documented way. And I think we're ready to move forward.
Great, thanks. All right, well, seeing no more questions, I think we can do the feedback survey and be on time. Thank you.
Thank you.
Thank you.
We also have another break between now and 2 o'clock Eastern time and 11 o'clock Pacific time. So if you could all be back by then, that would be great. Thank you. So I think we're ready to get started with the next project. Scott?
Okay, well, this is very good. Welcome back, everybody, from the break. So our next talk is going to be from Song Lee from UCLA, talking about the immunomodulatory periodontal microneedle patch for tissue regeneration. So take it away. And we aren't hearing you yet, so.
Okay, got it. Thank you, Scott. So first I want to briefly introduce our interdisciplinary team, including both engineers and dentists. In here, this team, we have scientists and grad students who did most of this R&D work. We also have collaboration with faculty in dental school. In addition, I want to also thank Dr. Ben Wu for his great support and guidance regarding the development of the product and clinical relevance. Next slide, please. We have heard that oral diseases affect billions of people. And here we are trying to address is periodontitis, which is a destructive inflammatory disease and chronic infections in the periodontal tissue. So this could cause the degeneration of the bone in gingival tissue around the teeth. In the current therapy, mostly is antibiotic treatment to suppress bacterial infection. However, this is not sufficient because in this case, the direct destructive cell types are the immune cells from our own body, including macrophages and T cells. So people have tried to promote the periodontal tissue regeneration, but a direct regeneration strategies may not work because in this highly inflammatory environment, even though you deliver growth factors, etc., it may not be effective. In addition, you will require a very high concentration of growth factors such as BMTF or BMP2 to achieve certain regeneration, but yet immunomodulation for cytokines. In most of cases, you need only a 1% or even lower concentration of these growth factors. So our approach is to take a fresh look and address the root of this problem with two kind of components in our drug delivery system. One is antibacterial by delivering antibiotics, and two is to trying to tune the pro-inflammatory signals in this microenvironment into pro-healing signals. In what we developed is a product as a microneedle patch could have a sustained delivery of the drugs. So this microneedle patch will achieve a painless delivery. It's suture-free, it's biodegradable, and allow a sustained delivery, and which is tailorable for a couple of weeks. And we, in our previous study, we have delivered antibiotics to suppress inflammatory signal. In addition, we can also deliver cytokine as well to modulate macrophages from pro-inflammatory to anti-inflammatory. And we have published the previous work in the Cell Press Journal Matter, just accepted for publication. And for the translation of this technology into a real product to make it easier for FDA regulatory pathway, we have looked into all of the drug candidates and simplify the design of this microneedle and formulation. So in this case, the microneedle is designed as a biodegradable, and we use the polyvinyl pyrrolidone as the microneedle component, which is water soluble. So upon delivery, it could be dissolved quickly within minutes. And inside, there are PLGA microparticles containing an antibiotic, azithromycin. And the reason we chose that is that because this is not only an antibiotic, but also has anti-inflammatory effects. And this could achieve a twofold twofold effects on the in this microenvironment. So far we have done, in the past 8 months, we have done in vitro study to demonstrate this drug delivery system can suppress bacteria in vitro, and also you could suppress the inflammatory cells. It's biocompatible in vivo in the rat model. And currently we are doing a long-term study in the next 2-3 months in rat model to try to prove that indeed we could achieve the regeneration of lost periodontal soft tissue in bone. In addition, we probably want to do some short-term study to demonstrate the biocompatibility of this product in a large animal model, in a mini pig model. For the regulatory path, it will be a new drug in combination with biomaterials, with 505B. For future potential business development, it could be a partnership with large dental companies in the local area, or it could be a startup. And we have a team interested in this startup approach as well. So this slide summarizes some of the recent findings. On the left, you could see some of these particles fabricated in our lab. And these particles are packed into these microneedles. In vitro, you can see, by the way, this is ex vivo delivery of this microneedle into the tissue. You can see this patch membrane is still here. It's not completely dissolved yet, but this part of the needle is already inserted tissue. That will allow multiple sites of delivery. You could have many of these small sites in the periodontal tissue delivering drug and overcome the transport limit in the tissue. In vitro, in the bacterial culture, we demonstrated this microneedle patch can release the drug and suppress the bacterial strains relevant in periodontal disease. We also tested this in vitro with the human immune cells, including macrophages and T cells. And here is just an example of in vitro study, how these microneedle released chemicals can suppress macrophage release cytokine, including tissue necrosis factors and interferon gamma. And here just shows that untreated sample, the figure resolution should be better. And also, if you use particles or microneedle patch alone without drug, you don't see much effect. But if you have soluble azithromycin or microparticle release azithromycin, microneedle release azithromycin, you can see a suppression of these inflammatory cytokines. In vivo, we have evidence in short term, this is just short term study to show that indeed, if you compare 4 groups, healthy, sham operation, microneedle patch without drug, and microneedle patch with azithromycin, we can see antibacterial activity with the release of azithromycin. Also, we can have suppression of interferon gamma, TNF alpha, in interleukin 1 beta as well. And we also observed some improvement of release of this regeneration signal. Next, please. For regulatory update, so we have decided this could be a 505B pathway, and we're trying to develop this as a drug device combo product. We're also preparing the pre-IND package for the meeting with NIH in the next 2 months. Next. For commercialization, we have filed an international patent at the end of last year, and we probably will provide some supplemental IP to update the patent application to include more information. We are working with DOCTRC team, regulatory team, also UCLA Technology Development Group to further develop this IP.
We'd better move on. Do you have can you go through the rest of your slides quickly?
Just one, maybe 30 seconds.
Okay.
Next. So manufacturing, we have identified 2 CMOs. We are going to have a partnership with them to develop scalable production. Next. For the next 6 months, as I mentioned, for research, we're going to finish the long-term regenerative studies in rat model, in short-term biocompatibility work in mini pigs. And regulatory is 505B pass for commercialization. We want to work with some MBA students to develop a better business plan. And for manufacturing, we are trying to scale up this production process. Thank you.
Okay, well, thank you. I think Kay Fuller and I have a similar question, which is that the patch is actually a delivery device, and then you're introducing a drug nanoparticle. And so are you planning to meet with both device GMP and drug GMP requirements in your combination, or how are you going to strategize this?
That's a good question. It will be a drug device combo in this case. We have actually experience for another product, a drug delivery system. We had a pre-IND meeting with NIH already. In that case, it's just a very simple configuration of the polymer delivery system. I think we could get through with the regulatory issues.
Okay. And William Giannoble asks about the use of the microneedles. And I guess the 2 questions, how do you categorize the use of an antibiotic as a regenerative approach? And how do you target the microneedles to the intradental region where the bacteria reside? And I guess also the question I would add to that is, how do you deal with the fact that in biofilms, the bacteria are much more resistant to the antibiotics?
Yeah, these are good questions. For the antibiotics, there are many of them, but we did an in vitro screening. We found that erythromycin not only can suppress bacteria, but also can suppress macrophage, the inflammatory signal. So that's the difference between this antibiotic and others. We yet to test whether it could work in the rat model. In the past, we did design deliver interleukin-4 together with these antibiotics. But in this new model, we think the antibiotic erythromycin alone could achieve better regeneration. We're going to test that in the next few months.
Okay, and does it spread far enough from the place of insertion to the place where you need the action?
Yeah, I think this is an important point as well. And it's also the difference between the microneedle and microspheres. The microspheres, unless you do direct injection into tissue, will just stay in the pocket between the teeth and the gingival tissue. For this microneedle, this has multiple spots. If you have, let's say, tens of the spots at different sites of the tissue, you overcome this transport issue of the drug in this soft tissue. You really have many small release centers of the drug as well. So there's a difference between the microneedle approach and the microsphere or nanosphere approach, simple injection.
Okay. And you have yet to see if it's increasing regenerative activity. I think that was a key part of the question before.
Yeah. In a short-term study, we haven't done a long-term study. In short-term studies by PCR analysis to examine the gene expression in the local gingival tissue, we have demonstrated several regenerative genes expressed, including BMP2, etc., in the local tissue. And we are going to hopefully show that in the next couple of months that in the long run, we could achieve that.
Okay, so we'll have to wrap up there. Thank you very much for your presentation. And I think we have our moment of silence for those to do their evaluations.
Okay, we ready to get going with the next one?
We are.
Okay. The next ITP presentation is going to be given by Brad Ament, who is a professor of anatomy and cell biology, director of the Craniofacial Anomalies Center at the University of Iowa. The title of the project is microRNA mediated alveolar ridge preservation and restoration. This is also a CDOCTR stage 2 ITP that's being considered for transition to stage 3. Take it away, Brad.
Thank you. So we have a company called Nature Mirai that we're using our new technology with. So we have a great group of surgeons, orthodontists, and bioengineers that we've assembled to help us with this. Next slide, please. So as we've shown today with great presentations, there's a great need to rapidly repair and regenerate bone naturally, and we want to do it without bone grafts or invasive procedures. So we want to grow new dense bone that integrates with existing bone using gene therapy. So we're going to do this in children, adolescents, and adults. We can do both large critical bone defects and small lesions. And so we're going to do our first human clinical trial that clearly focuses on safety and efficacy of our product to promote elevator bone regeneration for better dental implants. So what's our product? It's a plasmid-based microRNA inhibitor system. And so we can modulate microRNA activity, and we can use this to reprogram cells and have cell differentiation and enhance bone regeneration. So it's a lot better than oligonucleotides and integramers that are out there. It's not toxic to cells, and most of these that have used these oligonucleotides and integramers, they have not made it past phase 2 clinical trials, and they're toxic and have off-target effects. So ours is very different. So we'll apply it to a collagen sponge or an osteosponge or osteowrap and insert it into a bone defect, and we're looking at a 2-socket. And so we can also combine it with a non-toxic delivery system. But the problem with the oligos is you have to use a toxic delivery system. So we've already filed an FDA IND application. It's on clinical hold, and they came back with several things that they wanted to be addressed. And we've addressed most of these now. We're working with the FDA, and we want to finish our application and get approval. And so again, we formed Nature Mirai, and you can look at our website. We can sell individual projects or mice, and we've started therapeutic applications. And we are partnering with other companies and sell our inventions, and we're providing technical support. So we've shown that we can apply highly purified PMIS miR200A plasmid DNA, it's naked DNA, to collagen sponges, and we can grow dense bone pretty quickly. And so the NIH RAC committee invited us out to give a presentation on this. And what they like about it is plasmid DNA, which the FDA will approve pretty quickly. So we've done dosage experiments and indicate that 1 to 5 micrograms of our plasmid DNA works best to regenerate the bone, and more is not better. It's not more efficient. Time course experiments have shown that new dense bone is generated within 4 to 6 weeks. And we have partnered with our bioengineering group for calcium carbonate or heat shrinking nanoparticles can also deliver our molecule. We've done hard tissue sections, reveal a new bone integrates with the existing bone. We get vertical bone growth without using mechanical supports. And we've demonstrated that a heterogeneous population of cells will enter our sponge and take up the DNA within a day or two. And it's highly expressed, and then they reprogram these cells to form bone. We've done a toxicology test demonstrating that it's not toxic to the cells of the animal. And we've done an IRB now with our clinical expertise and folks that we have. And we're obtaining human patient tissue samples from the clinic to test then for microRNA expression and to see the effect. So all milestones have been met as outlined in our proposal. And I don't have any data slides to show. Next slide. So our FDA IND, and this is our project for plasmid DNA expressing our microRNA, our inhibitor. in a collagen sponge. And so we'll regenerate bone. And we can also inhibit inflammation. And again, this is a safety trial, which is what we do first. What we also show is that we can inhibit inflammation, and also we are reducing tumor formation. We have lots of applications for this system, but we're going to show safety first. So the FDA requirements they want us to do is, and we've recloned our plasmids into new kanamycin-resistant vectors. Sequence has been approved. Our clinical trial parameters have been approved. We did large animal studies, dog studies that they wanted, dosing, toxicology has all been approved. And the bioactivity and durability of observed effects have been completed. We have an NDA with drug development resources in Cambridge and also working with CDOCTR as well to get FDA approval. We're really wanting cGMP manufacturing now of our plasmid DNA vectors. And we're working with Pearson to do this so we can get this into patients. Next slide. So our commercialization manufacturing. So we have products that we do sell on naturemirai.com. We've done market surveys, have been completed, and they support the need for our system. And we've added new technology and industry experts to the company advisory board. So we've been awarded proof of commercial relevance funding from the state of Iowa, demo and gap funding from the Iowa Economic Development Authority. We've won first place elevator pitch. We're working with many groups. What we need is we have to purchase our IND ready plasmid DNA. We may have to repeat some experiments. Working with a commercial supplier for collagen sponges, and then we'll get our revised FDA IND application in. And then we're looking at our human clinical trials and get some funding for that. Next slide. So our plans then, so we do know that our inhibitor to miR-200A can regenerate bone, efficiently and safely, and there's no toxic effects. And so on that, we're just going to keep doing some more dosing experiments and some more large animal experiments. So the FDA IND has been submitted. We've done most of the revisions. We're really just waiting on cGMP manufacturing and some funding to get that done. So most of the concerns have been addressed. And well, then for commercialization, the company that we established, and like I said, that was a lot of work. We commercialized our PMS system, and a marketing firm produced the Nature Mirai qualitative report. So we're just looking for investors at this point. We're working with Pearson to make our FDA ready plasmid DNA, and also manufacturers of the sponges that we can use. And again, we have the IRB approval, FDA consultants identified, toxicology is done, and we're pretty much ready to go. And just looking for more people to work on a project. And of course, we support DEI. And that's my presentation.
Thank you, Brad. There's a few questions in the chat. The first one asks for maybe an additional comment or detail about the vertical bone growth and how you determine what's clinically significant.
Yeah, so we've done a lot of, like I said, the dogs and the rats that we worked on. And so using our controls and our inhibitor system, we can really see when you look at the alva ridge, and we measure that, if we can bring bone up to the level of the alva ridge. And a lot of times if you have a 2-socket and you just do nothing, of course you can grow bone, but there'll be that nice little dip in that between the teeth, if you look at that. And then if you want to do implants, it's really hard. Sometimes you have to repair that bone, but we just see the bone, the level of bone come right up to the alva ridge level. And we think that will be sufficient in a clinic setting.
Okay, great, thank you. The next chat question was about toxicology studies and what the FDA's requested.
Yeah, they wanted to see what we're looking at. We've done, of course, the bilirubin, the ALP, ASP, analysis, and also look at some cytochrome stuff. But also when you look at microRNAs, you know, some of the things in the liver toxicity, so you can activate some microRNAs that then activate some enzymes in the liver. And so we've shown that that's all negative. And our system is so specific that we're only targeting a microRNA that we want. And so we're not inducing or causing any other type of side effects that they see with using oligonucleotides, because oligos will activate all sorts of stuff in the liver.
Okay, the next question was about collagen sponge and experience that might not be ideal. Are there thoughts about an injectable product, perhaps with a collagen carrier?
So what we found is that we started using a collagen sponge because the FDA approves it. And so we wanted to be able to get in to our human clinical trials with something that's already approved by the FDA. The collagen sponge acts to actually sequester our plasmid DNA and kind of stabilizes it a little bit until the cells come in and take it up. And they do take it up, and we see expression, you know, pretty quickly by several different types of cells on the sponge. But we're working with other types of systems, delivery systems, and we're willing to partner with anybody that has any other types of matrices that can be effective.
Great, thanks. I see Pat has her hand raised. You want to ask a question, Pat?
I believe you're muted.
I had a question. You indicated that you put your microRNA DNA into new vectors. So is that going to, have you completed the necessary preclinical work with the new vectors, or do you need to do preclinical work given that essentially you've got a new vector system? How's that impacting your efficacy, you know, all the studies that you need to do that you'd perhaps done with your previous?
A good point. And yes, we have. We've done we repeated some of those, and it's the same. And but we'll have to probably repeat more, and especially with the CGMP. But the thing with the new vectors is that we just removed some of there was a few retroviral sequences in there, things like that they wanted us to remove. And so we made the vectors actually smaller, and so they're taken up easier by the cells and expressed. So we see no difference in expression levels or anything like that. But yeah, so now they clearly won't be toxic to the cells. But we repeated some of it, and there's no difference.
I mean, that's good. It's just you know how administrative are they going to be about it in asking for repeated studies is all.
Yeah, the FDA likes our approach. And for one thing, because of the plasmid DNA. So I think that they're excited about it, is what they tell us. And they think that, you know, we can move this forward pretty quickly.
Excellent.
Thank you. Another question was related to competition that might be with simpler products. And have you any sense of the competitive advantage you may have and how that weighs with potential partner perspectives of your technology?
Yes, and we've done this too, and our marketing survey addressed this, is that there's nothing like our system out there. And so we spent many years developing this system and testing it. And we have mouse models. So it's the only system that we can make a mouse model with. And it's so specific that we can then knock down, you know, a microRNA within a cluster that you can't do with CRISPR-Cas or any type of gene targeting. So, but we have this is the only one we have, and we have multiple patents on this and also in the process of this. So it works so much better than anything on the market. So, but I'm sure as other people see this, they'll come up with something.
Okay, thanks, Brad. So we'll have to stop to allow time for comments to be entered electronically. So you may want to address some of the remaining questions either via the chat, or I think you'll have an opportunity to do that via a website interface later. So thank you, Brad, for your presentation, and thank you all for your questions.
Thank you, everyone.
Hi, Bill. This is Stephen. Let me know when I get started. And I have some scroll out animations. So all right, I can tell somebody to advance those.
Okay. So the next presentation from Greenmark Biomedical is targeted remineralization treatment using mineral-loaded starch nanoparticle. Steve, go ahead.
Well, hi, everyone. I'm happy to start right on time, appropriately at 2:30. Ouch. So this is our Greenmark U of M ITP team, including co-inventors, Dr. Nathan Jones, Brian Clarkson, and Jörg Lahan, and our team at the School of Dentistry, and last but most important, my spouse, Dr. Wendy Bloomberg, and my thought partner. So the Greenmark leaf logo symbolizes a combination of our bio-based platform with targeted delivery, meaning our tiny particles go exactly where needed, and they bioresorb after the job is done. Now you can see our tree at Greenmark has grown substantially, now including quality management, customer service, and sales functions. Next, please. Now, while surgical treatment unnecessarily destroys dental tissues, current non-invasive approaches are suboptimal in that they incompletely repair caries lesion. Next. We are developing two Crystal Care products, a fluoride-free and a fluoride plus version, and both are designed to, number one, treat dental hypersensitivity, or DH for short, and number two, provide more natural repair of early dental caries using a non-invasive treatment to help improve long-term oral health outcomes. Next. Now, the particles are produced from food-grade starch, and our research has shown caries lesions are negatively charged, and therefore we functionalize them with cationic targeting groups and load them with calcium and phosphate, the essential components that make up enamel. Now, upon diagnosis of one, DH, and two, early caries active lesions, Crystal Care treatment is applied in office with additional follow-on applications as necessary. Degradation in saliva then releases the minerals for formation of hydroxyapatite-like crystals. Next. Our products are for professional in-office use to be applied after routine dental cleaning to teeth with hypersensitivity or non-cavitated caries lesions. They are indicated for treatment of patients 6 years of age and older. Next. Now, we have confirmed dental occlusion and mineral deposition and are repeating this for our final products in Q1 of 22, which due to COVID is the earliest the third-party lab can get this done for us. Now, in past presentations, we have shown superior subsurface remineralization on natural lesions compared to a conventional product with equivalent fluoride dosage. We also demonstrated reduction and in some cases even disappearance of unesthetic white spot lesions when the subsurface of enamel is restored. Show a proof of principle for that. Now, current studies on extracted teeth are focusing on optimization of treatment cycles. Next. In terms of safety, internal tox testing passed, and we fully expect a third-party biological risk assessment to confirm safety. Next. Now, having vetted our strategy by a number of experts, we will register the fluoride-free product as a Class 1 medical device and the fluoride-containing product as a 510K Class 2 medical device for a DH claim. Now, after FDA clearance, we intend to follow this up with a de novo application for early caries treatment, claiming physical filling of lesions with minerals, meaning to structure regeneration. Next. Now, we plan to launch the fluoride-free DH product in the US market as soon as possible, and we will sell via the direct sales force we have developed for our Lumicare product. Upon FDA clearance, we will launch the Fluoride Plus product as a premium product. And we plan to leverage the fluoride-free product for our market launch in Europe. Next. Now, regrettably, after 6 months of working with a company that produces single-use 2 compartment packaging we were all excited about, they were severely impacted by COVID and unable to deliver for us. So we identified this in prior updates as a critical task for the BRA and shelf life studies. A key requirement is to package Crystal Care as a dry powder. This is in order to prevent premature release of its calcium, phosphate, and fluoride components. Next. Fortunately, we were able to identify a packaging partner in the meantime who is utilizing one of their existing varnish packaging lines, placing our powder in the well and including an ampule for water, plus a brush applicator used to mix inside the well. Next. Meanwhile, Nathan and I developed a non-dusting dry formulation that instantly disperses and forms a gel within 30 seconds. Next. We also proved that the mineral deposition consists of calcium and phosphate by EDX analysis, and we confirmed mineral deposition by SEM as shown on the next slide. Now, the predicate for our Crystal Care Fluoride Plus product is Relief ACP Gel, and ZenCP Plus is a calcium phosphate product currently on the market that is registered as a Class 1 device with FDA. We aim to launch our fluoride-free product as early as Q2 of next year. And clinical data obtained in the US would help launch the product in the EU market, where fluoride-containing products are coming under increased scrutiny under the new regulations mandated by 2024. After we secure FDA clearance for the Fluoride Plus version, we will launch the product in the US. Ultimately, we want a regeneration of enamel type claim for our Class 2 DH product after it's been cleared. And to this end, we plan to pursue a de novo application with the method of action being physical filling of lesions with minerals. Now, de novo has the advantage that it has no predicate, so we can finally speak to all of the unique advantages of our device. Next. A milestone 5 to be completed in Q1 of 22 is another example of delays caused directly by COVID as third-party service providers struggle to complete their work. Next. This October, we attended the first in-person ever event since February of 2020, the National ADA SmileCon show in Las Vegas. And our strategic advisor, Celerant, had their Best of Class Alley and the Digital Future of Dentistry events and enjoyed the largest footprint of the show, which included Greenmark as one of their 2021 Best of Class awardees. Greenmark was represented by 8 of our team members to help man our main booth and our Best of Class booth. We were busy throughout the 3-day event, performed lots of demos, and we generated many leads. We also, as you see in this picture, made our first sales. Very exciting. Now, if you go to the next, here we see show there was quite a bit of excitement for Crystal Care products viewed by all who visited our booth to become a game changer in preventative treatment. Meanwhile, Dr. Clarkson challenged us to prove out the robustness of our technology and to pause to determine the crystallography for both products. As a result, prior to clinical studies, we feel it's prudent to invest in additional product characterization in year 2 of the grant. Next. Now, we build our sales engine with the e-commerce capability, and we are in advanced discussions with 2 large global dental supply companies that value preventative care. And we're building our own sales rep network so the train is leaving the station. Next. We established a manufacturing product for the process for the LumiCare companion product and produced 160 gallons for our first packaging run in September. Next. By comparison, here you see the scale Nathan used during his PhD research.
We're going to have to get to questions.
Yeah, I have just 1 slide left, so I think we're done. This is the Crystal Care product process, and we've hired a chemical engineer to help optimize the recovery and purification. And if you go to the last slide, no need to discuss the details here, but this is the project plans for the next 6 months that the leadership team requested. And then if you can do one more advance, I'd just like to acknowledge our technical team active at our Accelerator Lab facility. Thank you all for listening. I'm happy to take questions.
Thank you. So exciting progress. Questions from the group? So what's the uptake on the Lumicare? What's your expectation for the ramp up of that?
So we are currently in the soft launch phase, as we call it. We just finished training our sales reps, and we are working in clinics to prove this out more fully. And really, next year is our big year for the Lumicare product, going from currently we have 10 reps, going to 22 reps by the end of the year. But as I said, we're talking to 2 large global dental supply companies. We are in advanced discussions with them. We visited both, one in Europe, one in the US, and they're excited about taking over the sales. And that's something we could transition over to one or both of the partners.
And displacing or getting market share for current dental hypersensitivity products. How do you see, what's the pitch on that?
So it's really primarily a regulatory strategy where we just try to get to market as quickly as possible. Most dentists know that all of these varnish products, fluoride varnish products, which are all for dental hypersensitivity, 510 class 2 device, they're used off-label extensively. So we're banking on that. The power of KOLs is they can talk to this while we as a company can't formally talk to it. The KOLs can speak to the uniqueness of the technology, get it out there, get more and more clinical data, and then use that clinical data to reposition the product for de novo application.
Thank you. So Pat Olson has a hand up.
Just out of curiosity, how do things like this get reimbursed, or do they get, it doesn't sound like it should be maybe that expensive, but on the other hand, you know?
Yeah, great question. Thank you. So the Lumicare product is harder because it's a test. There is a code, but most insurance companies won't honor it. But on the other hand, the treatment, that's where it gets exciting. Per tooth, there are several codes, D1354 and 1355 is the one that was just introduced in January this year. So there are definitely codes there. And the challenge is, of course, ultimately to be more or less regarded as a standard of care, where we will see much more significant insurance reimbursement and usage.
Right, we have a couple of questions on the chat. Do you have nano hardness data on clinically treated remineralized lesions? What are the environmental parameters that drive variability in the mechanical properties?
Yeah, great question. So sorry, we didn't have much time to show all the previous data. Turns out that sort of surface microhardness is not all that meaningful. Working with the group at Pittsburgh School of Dentistry, we utilized their team to evaluate micro CT, and we evaluated the subsurface of the lesions. And we showed essentially that a fluoride treatment or the control doesn't do anything substantial on the subsurface, but our fluoride-free, both our fluoride-free and our fluoride-containing products both preferentially restore the subsurface of the lesion. So we're measuring basically bone density or enamel density into the depth of the lesion using micro CT, and we were very excited about that. Frankly speaking, this is all on extracted teeth, of course. So it's all going to be up to the actual clinical studies eventually to prove this working in patients' mouths.
So real quick, a couple seconds. You mentioned hydroxyapatite-like mineral. Can you explain in more detail how you characterize the HA and its dissolution rate?
Yeah, so we've done at least the confirmation that calcium and phosphate are there through EDX analysis, so that's good. The next thing is to go into wide angle X-ray diffraction, really get some crystallography, because fundamentally hydroxyapatite or fluorohydroxyapatite differ quite a bit. And then, of course, depending on the ratios and how it nucleates or what exactly the mechanism is, we may be surprised to find an unusual crystal structure. But in all likelihood, it's going to be the most preferred formulation that's there, or crystal structure for hydroxyapatite or fluorohydroxyapatite.
Great, thank you. Mitsumi?
Great, thank you, everybody. So we are going to take a break right now and return for the next session, which is a breakout session. So if you could all return by 3:15 Eastern, 12:15 Pacific.